Breathtaking.
Inspiring.
Exhilarating.
Unique.
You are all winners here today in Berlin.
Very energized and excited.
It's such fun. There is dynamism and energy and enthusiasm and great innovation.
We listen here to 3-minute presentations from people from all over the world.
Our jury members have 0 to 5 points to give.
We have the winners of the labs from around the world, so we're dealing with the creme de la creme. And it's very difficult to pitch them against each other and figure out.
I want to talk to you about rhythm, heart rhythm.
How will you feel if you can breathe freely?
The power of developing software lies in the hands of just 1% of all people.
I don't think there's a better networking or learning opportunity that I've ever been a part of.
You get to meet people from all around the world, from very different backgrounds, so the inputs that you can get are massive.
Despite the fact that I'm so well traveled, I still met people here from nationalities that I have never met before in my life.
The biggest highlight is delivering a speech in front of 200 plus people. Another one is learning about research opportunities in Germany.
I will never forget these moments. I'm enjoying myself so much. I'm so grateful to be here. It seems like a great opportunity and it's unforgettable.
It's not only making the walls full, but it's changing lives for the better, and I absolutely love that. It's nothing better.
Can I start now, or shall I wait till 11:30? Tēnā koutou I rō rangatirama, e ngā mana, e ngā reo, ko tai mai nei, e ngā hau e whā. Nau mai, haere mai, whakatau mai rā. Ko te kaupapa nui I tēnei rā, ko whia te ārai ka tūrakina. Ke ngā kai whakawa, ke ngā kai whakataitai, kia kaha, kia toa, kia manawa nui. Tēnā koutou, tēnā tātou katoa. Welcome everybody. What I just said was welcome to you all, welcome to distinguished speakers, to guests, and of course to the important question of the day, which is the next wall to fall. To the presenters and the jury, be strong, be brave, be persistent, and most of all, I hope you all have a great day and try and enjoy the day as we go along. My name is Troels Petersen. I'm the MC for today. And as you will notice, we have quite a strict program. I hope you all have one of those wonderful programs that are lying up by the table there. My first job is really to take you through some health and safety, which is not the most exciting thing to do, but nevertheless, if there's a fire, the fire alarm will go and we will all evacuate through the exits there, over there, and we'll meet out on the parking lot out here. If there's an earthquake, drop, cover, and hold. And when the shake stops, if safe, we again meet out on the assembly place here. This is a very safe building, so feel assured that nothing will happen. You can see you look around, you don't really believe me. And the Wellington Emergency Management Services are just around the street, so we will be in safe hands. But I think more expected emergencies could be we need the toilet. And the toilets are through the hallway and your last door on the right. There are the Whakapuka. I think that is what I had to say about the health and safety. Again, I just want to wish you all an amazing day. And I will start out by presenting our first speaker of the day, which is the president of the Royal Society Te Aparangi, Brent Clothier.
Thank you, Trolls. Kia ora tātou, ko Brent Clothier tōku ingoa. I'm the president of the Royal Society Te Aparangi, and welcome to Falling Walls 2023. Nau mai, haere mai to our whare, especially to Ms. Winnie Switakowski, the Deputy Head of Mission of the German Embassy, and Ms. Deborah Jacobi, the Third Secretary. Our Falling Walls Association began with the German Embassy back in 2016. We've had a few hiccups of late due to COVID, so it's great to be back here in 2023 face-to-face with a cohort of 22 3-minute pictures from you, our participants. 20 from Aotearoa, New Zealand, fantastic, plus 2 from the University of the South Pacific in Fiji. Welcome, nau mai, haere mai. Especially thank you to the German Embassy for your continuing support and our judging panel who will be involved in making these assessments. The winner will travel to the global final in Germany on the 7th of November. So this is going to be an exciting day where we learn which walls are going to fall next. Kia kaha. I'll hand back to Trolls. Welcome.
Kia ora, Brent. Next, I would like to call Winnie Switakowski up and say a few words. Winnie is the Deputy Head of Mission for the German Embassy. So.
Thanks, Trolls. Guten Tag und kia ora. First of all, thank you to the Royal Society Te Aparangi for hosting this event. It's really great and a great job and an honor to be here today. It brings me a great pleasure to extend a warm welcome to all of you present here, the next generation of ambitious innovators and creative thinkers. In light of over 4 decades of scientific collaboration between Germany and New Zealand, your diverse research contributes to shaping and enhancing the world of tomorrow, and I really believe in what I'm saying when I'm saying this. One of you will receive the opportunity to participate in the grand final of the Falling Walls Lab and Science Summit in 2023. This provides a chance to become the breakthrough winner of this year. But more importantly, it gives you also the chance to showcase your individual research projects to acclaimed scientists and connect with these experts, opening doors for your future research endeavors. Spanning areas such as health, climate, and digitization, your research not only influences the present way of life, but also makes a pivotal contribution to society and coexistence in the years and decades ahead. It is no coincidence that the Horizon Europe program categorizes these areas as global challenges demanding our attention. Pillar 2 of Horizon Europe opens up the potential to further improve and advance the cooperation between German and New Zealand researchers. And for that, it depends on ingenious ideas like yours, ideas that have the potential to break through walls. The Falling Walls initiative got its name from the collapse of the Berlin Wall in 1989. The fall of the wall emancipated Germany and ushered in fresh opportunities for a nation to redefine itself. It is universally understood in Germany that walls can never be the path forward, whereas fallen walls paved the way for creativity, innovation, and liberty. You all have incredible potential. At the end of the day, regardless of who wins, you can all be proud of yourselves. Isn't it exciting that one of the projects presented here today might actually change the world one day? That is why all of us here present today are proud to be here and full of hope, and most of all, eager to listen to your ideas. I hope that the victor will enjoy an inspiring and worthwhile time in Germany, building networks and making new friendships. But for now, most important, good luck to all of you. Thank you.
Thank you, Winnie, for those inspiring words. My next job is to present the winner of the last Falling Walls event that we had in 2021. And that is Mohammad Rehan. He is not able to be here today, but he has recorded a small video greeting that we will play for you now.
Hello everyone, Kia ora and Assalamu alaikum. A very warm welcome to all the attendees of this event who are here to witness the diversity in research and innovation that is happening across New Zealand. You will observe that the researchers are trying to turn every possible stone to make this world a better place. My heart goes with all the finalists who have made it to the final stage of Falling Walls Lab New Zealand. All the finalists, it takes a great deal of enthusiasm and motivation to come out of our comfort zone and present the big idea that has a potential to break the wall. Let me assure you that presenting your idea will help you to polish it further, as this platform will provide you the feedback and help you make new connections and add collaborators to your invention. I wish you all the best for the final stage. Rock the stage and good luck. Thank you.
I was about to say thank you, Mohammed, but he can't hear me, so sorry. Before we get started, I just want to give a bit of history of the last couple of years. Obviously, during COVID, there wasn't much Falling Walls in 2020. In 2021, we managed to have an online event that was very successful, but it didn't really provide the same atmosphere for the participants as an event today. So what we have done this year is we have invited some of those participants down. And so there will be 6 of those present in the audience today. And we have given them an opportunity to talk a little bit about their project on the posters presented on the right-hand side over here. So if you have an opportunity in a break, then please take the opportunity to go and talk to them and discuss their research with them, so they also feel part of the Falling Walls atmosphere. Before we get started, I just want to start out by introducing our jury. The jury this year is chaired by Professor Phil Lester from University of Victoria. Phil is a professor in insectomology, and if you haven't seen any of his videos on bees and wasps, you really have to go and watch them because they are fascinating. So welcome, Phil, and thank you for donating your time once again. We also have with it Associate Professor Rhys Firestone. So Rhys is a researcher at the Massey University in social, cultural and health inequalities with a focus on young Pacifica people with non-communicable diseases. So thank you for being here today, Rhys. Associate Professor Alex Garishkin. Alex is at the University of Canterbury and he's our data expert today. Alex is really an expert in algorithms, biological data, big data science, and genomic comparison using all this genetic data. So thanks, Alex. Veronica Meduna. Veronica is a science reporter extraordinaire. She is currently editing The Conversation, if any of you are familiar with The Conversation. Before that, she ran a program of Radio New Zealand called Our Changing World that really portrayed researchers and the work they did. And the program is still going. It's not as good as it was when you did it, of course. But yeah, well, I didn't say that. But thank you so much for being here today. Then we have Monique Searches. Monique is the Chief Executive Officer of the New Zealand-German Chamber of Commerce. And I'd say she's a staunch advocate for everything relationship-wise between Germany and New Zealand. So she's really trying to promote interest for Germans to invest and experience New Zealand, and for New Zealanders to go to Germany and invest and or experience. So the other thing I should say is Monique has been with us right from the start. So thank you very much for taking this time, Monique. It's a real pleasure to have you here once again. Of course, my thank yous again to the German Embassy for your support of this event. And I should also say that if you read the program, our last jury was Chris Carmier Inslee. Unfortunately, he was unable to make it today, and we were not able to find a replacement with such short notice. So that's why you only see 5 jurors today. With that said, I think we are almost ready to introduce the first speaker. Before we do that, I will just talk a little bit about the rules for those of you who are not so familiar with the program. I have to make sure that I don't run over time here. So this is a presentation exercise. Presenters have 3 minutes to present their idea, and they have 4 slides in total. There's a countdown clock over there that will turn red after 2.5 minutes. Just to remind the poor speakers that if they haven't finished up yet, this is about time. Unfortunately, we don't take any questions from the audience because we often find, I've been in places where questions could be very long. Let's just put it that way. So we only take questions from the jury. The jury can start asking questions after 2.5 minutes, which might be a bit disturbing, but that's how it is run in Falling Walls Berlin for the final, so we want to keep it exactly the same. But we probably only have time for 1 question, maybe 2, which will be my responsibility to try and manage that. So, so much for the setup, so much for the jury, everybody involved. It is now my great pleasure to introduce our first speaker. Our first speaker is Ashlee Berryman from the University of Otago, and Ashlee is breaking the wall on endometriosis.
I want you to imagine or remember a time that you experienced pain to the point where it felt like it took up a significant portion of your mind, perhaps interfered with your ability to complete your work. Unfortunately, this is the reality for around 120,000 New Zealanders living with endometriosis. Endometriosis is a gynecological health condition where cells similar to the lining of the endometrium grow outside of the uterus where they're not supposed to be. And these cells or lesions can envelop or infiltrate into surrounding tissues and organs, and in some cases come together to create cysts of the ovaries called endometriomas, which together are not surprisingly quite painful. Hence, one of the most common symptoms of endometriosis is chronic pelvic pain. And like many other chronic pain conditions, women with endometriosis self-report cognitive impairment. It's therefore surprising and concerning that there is such limited quantitative research exploring the cognitive functioning of women living with this condition. So, in my lab, we utilised a comprehensive cognitive battery to better understand the cognitive profiles of these women. Our preliminary analyses have shown that on simple cognitive tasks, such as reaction time and task switching, there are little to no differences between groups. However, when tasks become more difficult and complex, requiring higher order cognitive functioning, we can see notable differences between groups on these 3 tasks here. Women with endometriosis appear to show deficits relative to healthy control participants, And the extent of those deficits is greater for women with endometriosis and chronic pelvic pain compared to endometriosis without chronic pelvic pain. However, it would be naive to assume that this chronic pelvic pain in isolation is what's causing the cognitive deficits we're seeing, given the range of psychological and non-pain comorbidities also associated with the condition. So we've also mapped a network analysis here of those factors that might be influencing cognitive performance. And after controlling for chronic pelvic pain, we can see that depression, anxiety, stress, and self-perceived image as a result of an endometriosis diagnosis are the factors most contributing towards these cognitive impairments that we're seeing. These are important and we don't want to neglect them. However, we remain confident that the most significant influencer here is the chronic pelvic pain, which can lead us to our next step, which is to target these pain cognition pathways in the brain to hopefully, one, restore the cognitive functioning back to the levels of healthy age and education matched controls, or at the very least, alleviate the chronic pelvic pain that is experienced by 1 in 10 women across their lifetime in New Zealand. Thank you.
We've got time for a quick question from the jury, if you have one. Alex?
Sorry, I couldn't hear over there.
Do you know if there is a genetic component to endometriosis? And if so, do you see any value in genomic tests for tackling this disease?
Partially, there is thought to be a hereditary aspect to it. No such gene has been found. It's more so thought that it's caused by retrograde retrograde menstruation that occurs in utero. Yeah.
Any further questions from the jury? No, thank you. We'll thank
you.
Ashlee. And I forgot to say that the timer clock comes with these inbuilt remarks of hurrying up. So sorry about that. Our next presenter is Benjamin Durán-Vinet from the University of Otago. And Benjamin is breaking the wall of marine biomonitoring. So welcome.
So, we have a lot of marine biological threats nowadays. We have toxic bloom, viruses, invasive species, which, one example, for example, we went to 800 million USD losses in Chile in just 1 week, which led to 23 million of fish deaths, and also a civil unrest, which lasted for months because the aquaculture industry had 20,000 redundancy. But an early warning system can actually eliminate or mitigate the impact of these threats. So our solution is called CyDX, which means CRISPR-Cas-based bioengineering of emerging substantial marine biological threats. And this is in 3 steps happening. First, we use a machine learning approach. This machine learning approach basically allows us to design almost automatically from genomic data the guide RNAs. And the guide RNAs basically guide our programmable protein, which is Cas13, to an environmental DNA, which usually the biological threats are going to be in the environment. So they are going to shred DNA, and we are going to have DNA in the water, so we can actually detect it. And when we detect it, we are going to have a detectable signal with fluorescence, which basically is a peak. So we know this presence, so we know that we have to do something there. And then, when we have this platform set, which is already set, we can actually use this. Oh, this is not. This, which are chips, graphene chips. So basically, these graphene chips can detect a small change in voltage when the CRISPR-Cas detection are happening. So we are going to have an electric peak in the system. And we have a peak, we are going to have a detection. And usually electric systems are more easier to integrate into electrical system, which make them faster and better. So our advantage for now are high specificity and high sensitivity. We have results in less than 50 minutes. We have a robust detection, even in presence of inhibitors, and also our reaction are only 4 to 8 USD per reaction, which is really cost effective. And I want to stop in the high specificity and sensitivity because basically this technique is able to detect any of us in this room. A high sensitivity is basically 1 in millions or trillions of molecules. So it's high sensitive, so we can detect very early on when we have an infection or a bloom or an invasive species here in New Zealand or in the world, so we will be able to reduce the impact of this in social, economic, and more importantly, in our ecosystems. Thank you.
Thank you, Benjamin. Any questions from the jury?
Sounds really interesting. Thank you.
Can we wait for the second, because either way it's going to be ring the bell.
We'll wait for that.
Yeah, I won't be able to hear. Yeah, there we go.
Good. So do you have any ideas for the guide RNA now? Have you gotten down that path that would lead you to the level of sensitivity and specificity that you're
Yes, we are actually using a public database available in New Zealand and also the world, and we have designed more than 10 guide RNAs that are actually working and real. They are highly sensitive, and the machine learning approach also helps us to avoid potential of targets, and it's designed, the machine learning is designed to give us a high active guide RNA, so we are going to have a better pick.
What are they targeting? Can you tell me?
We are targeting Undari pinetifrana and Sarvella spallanzani, which are invasive species, but also in Chile I worked with Alexandrium catenella, which are toxic blooms.
Thank you.
Thank you, Benjamin. Our next presenter is Brittany Park from the University of Auckland, and she is breaking the wall of elderly surgical recovery.
"2,000 patients over the age of 65 undergo emergency abdominal surgery every year, and of these, 1 in 10 will die following this operation. This rate is twice that of younger surgical patients, which is especially concerning given our ever-ageing population in Aotearoa. I work as a doctor in general surgery. We know that older patients take longer to recover following major surgery and have higher rates of complications. Current risk prediction tools in the setting of emergency surgery are not routinely used because they require a large amount of information and are hard to interpret. I want to find a way that is straightforward to identify which elderly patients are at highest risk of having these adverse outcomes using sarcopenia. Sarcopenia is muscle failure, primarily caused by aging, but also by cancer status, coexisting illness, and other factors. We are measuring sarcopenia in 2 ways that are practical in the emergency setting. First, as reduced muscle mass on the patient's abdominal CT scan, which is routinely captured as part of their diagnostic workup. Our meta-analysis of international studies has shown that reduced muscle mass in this way is correlated with adverse outcomes. The second way is muscle strength, measured by hand grip strength. This is a novel technique which is easy to administer and use by clinicians and easy to interpret. It's also non-invasive and inexpensive. We've recruited 90 participants into this study at 2 hospital sites in New Zealand, and we've measured their muscle mass and strength at the time of their operation. We are following these patients up to 1 year, and I'm using this information in order to create a risk prediction tool that accounts for age, sex, and ethnicity in order to classify these patients as sarcopenic or not. We're planning to use this on a digital app in the clinical setting. This risk prediction tool can identify vulnerable patients so that clinicians can institute bundles of care at the early stage in the recovery process. This will enhance patient-centered recovery and optimize outcomes so that we can, for our elderly surgical patients in Aotearoa, thus breaking this wall. Thank you.
Questions from the jury?
Thank you. Great presentation. I was just wondering whether you could add a little bit about what kind of added care you might provide to those that you identify as sarcopenic.
Yes, so I guess when we identify them as sarcopenic, that might be a time for us to work with our medical colleagues, so have shared medical care and have early identification and treatment of complications, as well as things like supplemental nutrition and increased work with people like physiotherapists and occupational therapists in the hospital.
Any further questions from the jury?
Yeah, thank you. So, if I understood correctly, and correct me if that's not the case, you're looking at this one particular parameter as a predictor of the disease. So do you know if there are other parameters that might be helpful to look into, or there is just this one that is extremely correlated with the outcome?
So there are other things that people measure like frailty, but at the moment there are about 70 tools that can be used to measure frailty in this setting. Sarcopenia, measured as a simple measure of muscle mass and muscle strength, might just be an easy predictor that we can use to add additional information in an emergency setting when we want to make sure that we're making the best decision for that patient and we're providing the best care based on their functional status.
Thank you, Brittany. The next presenter is Bryann Avendano from the University of Canterbury, and the title is Breaking the Wall of Resilience Planning.
Welcome to Aotearoa, New Zealand, our beautiful land with stunning mountains, clear lakes, and forests that are pristine. But it's full of hazards. Floods, earthquakes, fires. It's full of hazards everywhere. And on top of climate change, it's really difficult to plan for a resilient Aotearoa. Our communities thrive. How we can plan with that much uncertainty? In resilience planning, there is a problem. We normally assume independency on factors that are really interconnected. Do we invest on barrier in the coastline to avoid sea level rise coming? Or do we invest on emergency skills to our people? Complex decisions requires complex approach. Let me ask, why plan if we can play? I create a game that it's called Playing with Uncertainty. Basically, we invited 6 participants to play a role-playing negotiation where they need to negotiate, discuss, fight, and kind of bring in the idea of what they are thinking on what's better to invest. A model in the background, it's telling them how are their performance, and also it's telling them if their decisions are made in a correct way in terms of the scenario that we play. Yes, we play a scenario of floods, we play scenarios of fires, we play different scenarios and different consequences. But most important, they are exchanging, literally put the hat of a policymaker, exchange a card, role-playing card of a scientist, exchange a role-play card as a community leader. And they all exchange perspectives, which is the most important thing when we're making decisions. Because basically, we're engaging communities in decision-making. I trialed this during my PhD with policymakers, actually, in Canterbury area, with industry people, and also with resilient experts. And what I found is that I'm going to show you the future of resilience planning. Let me show you the future of our resilient world. Thank you.
Thank you, Bryann. Any questions from the jury?
Sounds great. It's an interesting approach. Now, did the people that you tested on, did they have an obvious change in their behaviour, or what did they say? What was the outcome for them?
Thank you for that question. Basically, they were very curious that what they found and different from their perspective was mind-changing. And what they found is that when they're making decisions based on only their own perspective, they were biased completely, of course. But in terms of how we exchange information, we were wrong. We normally think that we just listen, but we should put in the hat, or we should exchange shoes to understand others' perspective. And when we play roles, we found that that's basically happening. I didn't go to the virtual reality part, but yeah, that will happen in the reality too.
Thank you.
Any further questions from the jury? Thank you, Bryann. Our next presenter is Christopher Erwin from Auckland University of Technology, who is breaking the wall of quantitative intersectionality.
Kia ora. For each of us, our lives are lived at the intersection of the different identities that we carry. For myself, I'm white, upper middle class, I'm a male, I'm also a new father, I'm an immigrant to this country. And as we know, the identities that we carry can bring social privilege, or they can be a social burden for us, right? They could be a source of oppression sometimes. Now, and some of us deal with multiple sources of burden according to their identity. They are multiply marginalized. Now, I study inequality, and there's a problem with a lot of quantitative research on inequality, in that it assumes, or it presupposes, that these different identities we carry are independent. Now, from a research perspective, that drastically oversimplifies the picture of inequality in society. And also what it does, it prevents the stories of those that experience multiple forms of marginalizations from telling their stories through data. So my solution to this problem was to develop a quantitative approach which takes differences in group outcomes and decomposes it into main effects and intersectional effects, or interaction effects. So say, for example, that we wanted to look at low-income Pacifica women. The main effects that we'd be looking at would be being low income, being Pacifica, and being a woman. And now the interaction effects would be simultaneously being a woman and being Pacifica, or simultaneously being low income and being a woman. Now, adding these dimensions to the analysis provides a richer understanding of the landscape of inequality in society. So what I have here is just a super brief example, and I won't do any numbers, I won't interpret anything like that, but I want to show you how this process works. So in the first table here, I'm just regressing the outcome on the main effect and the interaction effect. Now, these effects can be aggregated up to table 2, where you see the differences in outcomes by group. And the real power of this method shines in table 3, where we can zoom in and look at low SES, or low income Pacifica women, and we can say, well, yes, the main effects matter, being Pacific matters, being a woman matters, being low income matters, but how these different forms of our identities impact or interact with each other is also an important part of the story. Two things I'd like to mention really briefly before concluding. In terms of policy, this has a lot of potential, right? Because if we're developing public policies to ameliorate issues of inequality, in society, then this allows policymakers to identify the question more clearly and to target those funds. Number 2, this is a very flexible method that could be applied to education, criminal justice, and health disparities, for example. Thank you.
Questions?
Kia ora. Thank you for that. Just wondering if you can give us a pragmatic example of how this model can inform how to improve equality with these minority groups.
Sure. I think one basic example, and this research is actually being used right now to formulate policies to improve literacy and numeracy in Aotearoa. And I think there was some blanket policies in the past that maybe just tried to throw some money at Maori or Pacifica communities. But here, by identifying the fact that there is a gender component, there is an income component, as well as a cultural component, I think it's changed the way that we're making policies to target these limited amounts of funds to ameliorate inequality.
Thank you.
Any further questions? Thank you, Christopher.
Thank you.
Our next presenter is Debashree Roy from the Reddit Institute at Massey University, and the presentation is Breaking the Wall of Plant-Based Cheeses.
If you look at this nutrition label, what stands out most? Well, 0% protein, and it's basically a starch oil matrix. This is the nutrition label of most plant-based or dairy-free vegan cheeses that we find in markets today. Unlike dairy cheeses, most plant-based cheeses worldwide lack protein. Dietary proteins are an essential part of a balanced diet, and dairy cheese is a substantial protein source for consumers. But the younger generations are increasingly concerned about the environmental impact of intensive dairy farming, and they are adopting plant-based choices. But the current options fail miserably on nutrition, taste, and price. With the global plant-based cheese market projected to reach $5 billion, there's a growing demand for better options. But why is it so technically challenging to make high-protein plant-based cheeses? Because plant proteins inherently lack the functionality and versatility of dairy proteins, resulting in undesirable textures and taste. But imagine a world where the irresistible pleasure of cheese isn't limited by dietary choices. Introducing Tejesvi, our breakthrough solution. Over the last 2 years, we have worked on a patented technology that uses a combination of plant proteins with innovative texture development processes, creating a highly functional plant protein base and enabling formulation of a variety of plant-based cheeses with protein contents comparable to dairy cheeses. With support from Massey Ventures Limited and KiwiNet funding, we have successfully completed prototypes and industrial scale-ups using our technology. But that's not all. In consumer sensory evaluations, Tejesvi's 20% plant-based cheese was a hit, beating existing products in all sensory attributes. The opportunities ahead really excites me. Currently, we are in technology commercialization discussion with a global dairy player in our efforts towards making these products affordable and accessible to every household. Let's work together in breaking the walls around plant-based cheese.
Any questions?
Awesome. Sounds really, really good. Is this being developed anywhere? Other people must be looking at this sort of thing too. It's not being developed anywhere else. And secondly, is it difficult? Is it expensive? Is it, yeah.
Yeah. So answer the first question, yes, there are technologies which are coming up, but what we know that currently there are none which are in the market. And most of those technologies which are coming up, they are more through startups who are in this field. So yes, there are technologies, yep. Second, in terms of price, yes, currently these cheeses are expensive, but with our technology, we will be price competitive with the existing vegan cheeses, even with 20% level, and we are also targeting to be price competitive to dairy. Yeah.
Do you like it yourself?
Yes, I love it. I have also a prototype in my bag, but I'm not allowed to give it for tasting because of safety and other issues, because that was not a prototype that we could develop now and get the approval. But yes, if people love it, we have demonstrated it, so yes, there are many who loves it.
Disappointing, but okay.
Any other questions from the jury? Alex?
Well, I guess one of the main arguments for developing this technology is the environmental effect of farming and dairy and all that. So is there a robust way of measuring the environmental impact of this technology when it is developed at scale?
Yes. So that's already in our pipeline that we will need to do a thorough life cycle analysis from the sources, not just from the plant proteins, but from the sources, how the plants are grown, and to the end product. What we have seen through our initial analysis that it is because they say that the plant-based proteins or the plant-based in general are sustainable. So the process will be sustainable with the steps that we are using. But I think sustainability is not just about the process, but it's also about nutrition and price, because we want it for the mass market. So that's where also we are looking into. But yes, these things will be in pipeline, and we are considering these aspects. Yeah.
Where do you think would be the biggest impact on the environment from this technology if it becomes global?
In terms of countries or in terms of?
No, in terms of the environmental impact. So I guess from dairy, one of the main ones would be methane or...
Yes, so I think what we believe in our technology, when we are starting, there is no water loss. When we are starting, there is a water loss before, but when we are doing our technology, the proteins are getting hydrated, and during the process, there is no water loss. So the impact on water quality, I'll assume at this stage will be expected to be the biggest impact, but there could be other things which we will take into consideration.
Thank you.
Yep.
Thank you, Debashree.
Yeah, thank you.
Our next presenter is Emily O'Riordan from Boudica Scientific, who is breaking the wall of extreme weather forecasting.
Tēnā koutou. Forecasting the weather is extremely expensive. This top image up here shows a temperature forecast over Banks Peninsula, and each of these grid squares are about 4 kilometers. In order to make this kind of forecast, we have to solve a whole lot of very complex equations in every single one of these grid cells. This is really computationally intensive, requires the use of a lot of supercomputers, and in New Zealand, Met Service spends tens of millions of dollars every single year to produce images like the top one. Obviously, it gives us a gist of what's going on, but ideally we'd love to work with images like this bottom one. This is a 300-meter resolution temperature forecast of Banks Peninsula. And as you can see, it gives us a lot more localized detail of what's happening, and this is particularly important for extreme weather events. For example, if there's going to be extreme precipitation in one valley versus another valley, that can be the difference between needing to evacuate a town or not. Unfortunately, going from this top image up here to the bottom one down here is about 500 times more expensive. So doing that over the whole of New Zealand, it's completely infeasible, not just for the cost, but also the environmental impact of using these supercomputers for that scale of forecasting. However, I'm currently working with Met Service in producing a generative artificial intelligence model that aims to solve this issue. The AI model will learn how to go from the coarse resolution images, like the one I showed you before and this one up here, and use also some other static details such as topography. And it will learn how to create high-resolution forecasts that have a lot more localized detail within them. The outputs will be extremely realistic, as the AI model is going to be constrained using the physical equations that are used within the dynamic modeling. And it will also hopefully correct some forecast bias, as the AI model will learn from not only forecast data, but also real observations. However, the main benefit of this is that the AI model is going to be extremely cheap to run once it has been trained, meaning that we cut out all of that cost that's needed for the dynamic modeling in producing high-resolution forecasts. This means we can run the model multiple times for every single time step and get slightly different outcomes, and therefore we're much more likely to pick up extreme weather events and also pick up their locality. Being able to get a whole ensemble of forecasts means we are able to create better weather warnings and therefore hopefully save lives and livelihoods across Aotearoa, New Zealand, especially with climate change. Thank you.
Alex?
So I guess the idea is to economize on the compute time that is being spent on those partial differential equations that you've shown that are very expensive to solve, and use the AI that takes a lot of compute to train, but then for predicting it's easier. But with climate change happening and out of distribution weather patterns occurring in the future, how frequently do you foresee retraining that AI model?
Yes, so it will need to be retrained every so often. We will be particularly focusing on extreme weather events within our training, so making sure that we have subsets of those. And once it's been trained once fully, we hope that it will just require fine-tuning in the future. So it will need some more training, as you say, but it won't be quite as expensive as this initial training is.
Would you say it still makes sense, even if we train an AI model to solve those partial differential equations once and forever?
Well, it would be nice, wouldn't it? But I think, as you say, with climate change, there are going to be things that are outside of the domain of what we've seen. And yeah, getting them to solve the partial differential equations for every single place in the globe might be slightly too far out of AI's reach right now, but who knows in the future? Might not be far off.
Thank you.
Thank you.
Any other questions from the jury? Thank you, Emily. Our next presenter is Jihwi Jang from the University of Canterbury, and the topic is breaking the wall of nitrate removal via tree.
Kia ora koto. Today I'm going to explain about bioremediation potential of radiata pine to mitigate nitrate contamination, especially in agricultural area and dairy farm in New Zealand. As we know, one of the main national income of New Zealand is primary industry, but let me talk about some negative impact. So, excessive amount of nitrate chemical fertilization and urine patch from animal can cause environmental and public health concern caused by nitrate. This is because nitrate can be changed to ammonium, and after nitrification process, is highly likely to reach to groundwater. So I would like to suggest plant radiata pine tree to mitigate nitrate reaching concern, because radiata pine is most common exotic tree species in New Zealand due to plantation forestry, and it has been reported that their growth rate and higher biomass, even more than California, United States, where actually they are from. So after my grasshouse experiment, treatment of boric acid and sodium selenite can foster their nitrate uptake by time. So for this reason, I would like to suggest to plant radiata pine in the agricultural area with a small amount of treatment of sodium selenite and boric acid. Thank you.
Veronica?
Thank you very much for this presentation. Why pine? Have you tried other trees as well, or is pine mostly because it is already a commercial tree here?
So I want to try a kind of comparison study with some native tree species, but it has been reported that radiata pine is more growth rate compared with the native tree species. So finally I choose this tree. And also most common tree species, so most for bioremediation, I think is more cost effective rather than purchasing native species. That's why I choose. Yeah.
Any other questions from the jury? Thank you. Our next presenter is Jildou van der Werf from the University of Otago, breaking the wall of personalized cancer treatment.
All of you have cancer, and we give you the same treatment. but your DNA is different. So for example, you guys have genotype CC, you guys CG, and you guys have GG. Now, only you guys respond to the treatment we give you. That seems a bit unfair, no? We have a lot of exciting new treatments on the market. Like for inflammatory bowel disease, which is a precursor for cancer, we have drugs that suppress your immune system. And for cancer, we have drugs that activate your immune system, called immune checkpoint inhibitors. These drugs come with far lesser side effects. And for some people, they are the magic bullet, but others don't respond at all. How great would it be if we can predict who's going to respond by looking at someone's DNA? Because it might take a doctor months to figure out the right treatment, but it only takes a few hours to figure out someone's genotype. This is the wall I want to break. In our DNA, we have 1 million different places where we can have just a single letter difference, also known as a SNP. Now, most are insignificant, but some have a huge effect on how our body functions and how a disease progresses. I am mainly interested in the one in the DNA for IL-6, which is a cytokine from your immune system. On this spot, you can have the letter G or the letter C, and this is known to affect your chances for many inflammatory diseases, including IBD and cancer. So to test how well this predicts responsiveness, we created mice that are exactly identical, except for just this one letter difference. First, we induced inflammation in the mice, and we saw that the C mice got more inflamed. So then we promoted inflammatory bowel disease. And not only did the C mice get sicker, measured with, amongst others, weight loss, when we gave them drugs specifically targeting IL-6, they responded better. And this is exactly what we hope to find. Now, we have strong reason to believe this will work for cancer, because when we look at patients that don't respond to treatment, this SNP comes out as a factor most influencing it. And when we isolate immune cells from the mice, we see that they behave differently between the C and the G, which could explain the why. So I'm going to repeat these experiments in mice with cancer and figure out the best treatment for the right genotype. But after this, we can use it for all sorts of inflammatory diseases. This can lay the foundation for treating people based on DNA. Then we can say that when all of you have cancer, you guys might benefit from ICI treatment, and you guys need a different kind of drug. This shortens the process for a patient in finding their magic bullet. And after this, we can even use it for all sorts of inflammatory diseases. So there's so many more walls to break. Thank you.
Thank you, Jildou. Any questions from the jury?
This is your expertise.
Yeah, thank you. So with your example, with some patients responding to one sort of therapy and the other ones responding to the one you're developing, are you imagining some sort of genetic test before the drug is prescribed? And if so, what sort of genetic tests do you have in mind to choose between the existing drugs and the one that will hopefully come out of this research?
Yeah, that's a good question. So we just want to look for this specific SNP. And then based on this, with the experiments that we do, we can figure out what treatment works best for what genotype. And then when we look at someone's DNA, we can already see, okay, this treatment might benefit or not at all. Whereas now a doctor will just give you the treatment and then after 4 weeks maybe assess if it worked or if it didn't work, which is a valuable time gone lost.
But do you think it's realistic to test for a particular SNP in the clinic, test every single patient for that particular SNP in the clinic?
Yes, it's a very easy and cost-inefficient method. It's just like when you do a COVID test, it's also a PCR.
So it's PCR-based, cool. Thank you.
There are companies in the States that would love this sort of thing that for altering insurance claims and that sort of thing. So this person, I know they've got the SNP, so they're more susceptible to cancer. Are you worried about that? How would you, is it a concern?
What is your question exactly?
So there are companies in the US, for example, that insure, health insurance companies, that could use this sort of information to alter their premiums or their likelihood to insure people on the basis of their potential to develop cancer.
Okay, yeah, we don't suggest to test everyone for this, but only in the stage when you already have cancer, then to use it to determine the best treatment.
Okay, all right, thank you.
Thank you. Our next presenter is Kristin Bohm from the Institute of Environmental Sciences and Research, breaking the wall of wasting our waste.
Yeah, thank you.
Fun fact. Did you know how much poop a human produces per year? You wouldn't guess this, but it's actually almost as much as a panda bear weighs. Wow, there's really a lot of poo that goes down the drain and needs to be treated. In fact, 45 million tritons of this treated waste is produced per year globally. And although the treatment process is quite costly and the end product, sewage sludge, contains valuable resources, still, most of it we just throw out for nothing. So it goes to the landfill, or we discharge it to the ocean, or just burn it to the air. So why we are still wasting our waste instead of using other options? One solution I'm proposing today is black soldier fly bioconversion. This technology has the potential to create a circular economy by recovering resources and generating high-value products from our waste. In fact, we have shown that larvae of black soldier fly can feed on sewage sludge and can reduce it by more than 40% within 2 weeks. This is actually 200 times faster than natural decomposition on landfills and 10 times faster than composting. Also, we would reduce a lot of greenhouse gas emissions. More importantly, black soldier fly bioconversion of sewage sludge recovers resources and generates a high-value protein and fat-rich biomass. Products from this technology can be used in agriculture or other industries to make new products, which we can use again. So for example, the fat composition of the black soldier fly larvae is quite similar to plant oil, and some of the components have been shown to have antimicrobial activity. So one could potentially make some functional cosmetics out of it. So would you have imagined that you can make soap out of poo? I would say, let's break the wall of wasting our waste and implement other solutions, such as insect bioconversion, to use our waste more efficiently and to create circular economies. Kia ora.
Thank you, Kristin. Any questions from the jury?
Have you tried it? Is it working? Is there a system going at the moment that does this?
We don't have a pilot system, so it's still on lab scale. But on lab scale, definitely it's working. So I had the larvae in my hand. I put it on biosolids. I know it's stinky. By the end, they are growing, they are quite fat, and we reduce it. So it's like obvious numbers from my research. And this is really soap made of black soldier fly fat.
Wow, cool.
I have to say, it's not fat on the biosolid itself, but it's possible that you can do it.
Thank you.
Can I just quickly ask about the black soldier fly itself? Even in a pilot scale, it would have to be some sort of containment because they may not want to stay on the particular poo you provide?
Yeah, so people are quite risk averse, and I thank you for this question. The beauty of this insect is that only the larvae are feeding on the organic waste, not the fly itself. So you can easily separate the fly stage, where you think they might transmit pathogens, from the larvae stage. Also, the fly itself is actually not really feeding organic waste. So basically, you might not find the fly in your house and feeding from your own like sandwich or stuff like this. It's not as the house fly. This fly is really smooth flying and actually mainly around your compost bin. So it's kind of safe technology.
Any further questions from the jury? One more.
What do you think is the biggest challenge of turning this technology from the lab into the commercial space?
Thank you for this question. It's a good question. Obviously, there's a bottleneck actually to market your products. I think that's the most challenge. There's obviously a challenge to keep the flies happy and to mate and make a lot of eggs, but I think the most challenge is actually to explore the market and to do the social science and to talk with people and say, look, it's not a yuck factor. It can be used. Even if we just feed it on food waste, they're quite beneficial. So it's just to explore the market.
Thank you.
Thank you, Kristin.
Thank you.
Our last presenter before the lunch is Krzysztof Maliszewski from the University of Canterbury, who is breaking the wall of medical imaging limits.
We all know these portable devices for measuring health indicators, blood sugar and blood pressure. Imagine if there was a device able to detect diseases at their early stages, health problems on the rise worldwide, such as Alzheimer's, Parkinson's, or even cancer. There is a technology that can enable that breakthrough. It's optical coherence tomography, OCT. It is based on retinal images obtained by scanning the eye with light. But there are obstacles on the way to achieve this great goal. The current OCT devices are heavy, bulky, not fit for the at-home use. Only the best of them, meaning the most expensive ones, can reach the limits to image the diseases and detect them. But also they reach their limits when it comes to image quality. Our solution will break these barriers. It redefines the image generation process of OCT. In our solution, the low-performing imaging generation algorithms are replaced with an ultra-high-performing artificial neural network with no fundamental constraints associated with the standard image processing techniques. The raw data that comes straight from the OCT devices is transferred into much more detailed images, as you can see here. Being fully software-based solution, our solution can help with miniaturization of the devices and cost reduction of these devices. But at the same time, it will deliver much more detailed images never seen before. With the new OCTs, companies established with the very goal of creating portable devices, truly portable devices. Our solution will help people to see their health with their own eyes. Thank you for listening.
Thank you, Krzysztof. Any questions from the panel or the jury?
Thank you for your presentation. I was just wondering, could you fill me in a little bit more as to how those retinal images show early stages of disease?
So, in short, when OCT scans the eye with light, you can create an image of the back of an eye. Depending on the structure of the back of an eye, you can diagnose patients with diseases, whether they have something wrong in their body or not. The more detailed the images are, the more sure you can be and more reliably you can detect any inconsistencies, any problems in terms of the health.
Any other questions from the jury?
Do you know if those devices are used in clinics at scale for any of those Alzheimer or cancer? Or?
So when it comes to more complex diseases such as Alzheimer's, Parkinson's, or even cancer, there are studies made by researchers that show the possibility of detecting these diseases, they are able to do that. It is not at this point in time to do that in a doctor's lab. There are companies that are able to detect the diseases. You can do that online, but there are specialists for that kind of thing. But with the AI created that can improve the image quality, it will be easier to create another AI to detect the diseases and make it mainstream.
So I guess those will be the companies that can serve as potential customers for this technology if we are talking about commercializing it.
Can you repeat, sorry?
So if you think in terms of commercialization of this technology, who would be the customer? Those companies that you just mentioned or someone else?
So for us, people who create these kinds of tools, for me, it will be the manufacturers of the devices. We will be proposing them the licensing of our solution, or another way of commercialization is to create an application, web application, and then allow people to deliver the data, so we will be able to improve the image quality and maybe come with a collaboration with people who create AI for detecting diseases.
Thank you.
Any further questions? Thank you, Krzysztof. And hereby ends the first half, and what a fascinating series of talks. Thank you very much for your presentations. Thank you, jury, for some excellent questions. We now have a lunch break to 12:45, 1:45. Just want to ask everybody to make sure you make your way back into the room here about 10 minutes before that, so about 12:35, so we can start on time. So ready to start the next set of speakers, which I'm very excited about. And our first presenter in this second session is Lysea Haggie from the University of Auckland, who is breaking the wall of brain and body.
Mind and soul is entirely separate to the body. No offense to Descartes, but I completely disagree. Our intelligence, emotions, and consciousness arise from the billions of neurons in our brains, which are intricately connected to our bodies in a complex network. This neural network processes an immense amount of information. Performing a synchronized dance of communication, it enables us to think and act. Neurons communicate through changes in their electrical activity, sending Morse code-like messages between brain and body. But we still don't really know how to decode these messages or how they are produced. Understanding the relationship between brain and body is vital to treat disorders such as stroke and Parkinson's, as well as to develop new technologies like brain-computer interfaces and neuroprostheses. I am breaking the wall between brain and body by building multidisciplinary computational models using next-generation artificial neural networks. I have built a model of tens of thousands of neurons with hundreds of millions of connections between them to replicate the structure and activity in the part of our brain called the motor cortex. I then combine this with biomechanical models of the musculoskeletal system to be able to produce digital simulations of human movement. Using computational models is necessary to be able to investigate complex interrelated systems which are difficult to measure or perform experiments on. This approach will help us decode the language the brain uses to control our body. This approach can also be extended to other regions of the brain and other organ systems of the body to investigate their complex interrelationship. Emulating the structure and function of the human nervous system will also inspire new forms of artificial intelligence. Today's AI can interpret and generate text and images, but the next step is to be able to build physically intelligent systems that can interact with us and the world around us. Through a multidisciplinary approach, combining not only neuroscience and engineering, but also psychology, philosophy, and ethics, we can develop neurotechnologies that have the potential to transform human existence and benefit everyone. Cool. Thank you. Questions?
Thank you, Lysea. Any questions from the jury?
Thank you, very nice talk. I'm wondering, how did you collect your data? So basically, I'm just guessing, to train a model like that, you would need to be some sort of neuron firing data, so some readouts from the brain, as well as some movements that are happening to the human. So where is the data coming from?
So I actually used previously published experimental data of the neural circuitry in the cortex to define the connectivity in my neural network. So a lot of my project was to be able to define the connectivity to create and replicate the spiking patterns which are recorded from intracortical devices and compared that to also previously recorded spiking data of the neural cortex, of the motor cortex.
Right, cool, thank you. And can you also tell me a little bit about the type of network that you're using? So presumably there is an artificial neural net between the brain neuron firing data and the movements.
So I'm using a specific neural network called spiking neural networks, which are a bit more based on biological neurons, and they have an all or nothing response. So they actually have the temporal relationship of the accumulation of action potentials, and then they fire action potentials down the neuron. So they're a bit different to traditional artificial neural networks, and that's why I called them next generation artificial neural networks, because they have that temporal component.
Thank you.
Okay.
Just a quick follow-up question.
Yeah.
Thank you. Do you think that our ethical considerations are evolving in step with the innovation, the technical innovation here?
No, and that's actually a really important aspect that I think is really important for me personally, because there are such big implications of, I guess, what I called embodied artificial intelligence, which could displace a lot of labor and things like that. So I think that ethics is a really important part of this, particularly if, and you're dealing with brain data, which in terms of behavior and cognition, like that could also be ethically problematic of like trying to read people's minds, but we're a bit far off that. So the ethical implications are huge. Cool.
Any further questions?
Just from a transformational research perspective, where would you like to see this model? Where did you foresee this model to be in the future? How would you use it?
Ideally, it would be for like, kind of like brain-computer interfaces for like paraplegia. So for people to be able to interact who can't. So maybe they can control things like interact with their world that have that are disabled or can't move. But obviously, if you're someone like Elon Musk, so you're like, oh, that technology can also be used for turning your lights on or stuff like that. But I would prefer to see it used in a clinical and a necessary setting.
Thank you, Lysea.
Cool. Thank you.
Our next speaker is Matthew French from the University of Auckland, breaking the wall of breast cancer treatment.
Breast cancer is the most diagnosed cancer worldwide, contributing to 12% of cancer cases and 30% in New Zealand. DCE MRI is the gold standard diagnostic tool for breast cancer. During MRI, a contrast agent is administered which highlights the tumour location and its boundaries. In the face-up position, a standard MRI can be obtained. However, it is very hard to distinguish the tumour between surrounding tissue in a standard MRI. You can't take a DCE MRI in this position due to the nice setup you have in the prone position. So not being able to see the tumour in the face-up position is very difficult, and so people have proposed that image processing techniques be used to map the contrast enhanced information that you have in the DCE MRI to the standard MRI in the face-up position. However, standard image processing techniques typically fail to estimate the motion between face down and face up due to the large and complex motion that occurs in the breast tissue between these positions. Furthermore, clinical techniques do not highlight the tumour location and its boundaries in its entirety, and this contributes to 20 to 40% of patients having to undergo multiple surgeries. I'm proposing a hybrid biomechanics and AI approach to overcome the imprecision of current clinical techniques. So biomechanics approaches are the current state-of-the-art for this task. However, they can't account for all of the motion due to approximations made in the models of breast anatomy and tissue mechanics. On the other hand, AI leverages neural networks and extensive data sets to learn the motion and provide models which are efficient and can pick up components that have maybe been missed by biomechanics. So the ultimate goal of this approach is to have the biomechanics do the heavy lifting and then have the AI pick up the residual components. And so the impact of this will be to provide clinicians with a method of precisely locating the tumour in the face-up surgical position. And this is important because during breast cancer treatment, residual cancer tissue can be left behind, and in this case, reoperation is required. So the ultimate goal is to provide this method so that reoperation rates can be reduced.
Thank you, Matthew. Any questions for Matthew?
Thank you. So how far along are you with it? Have you developed this? Have you got some preliminary trials and that sort of thing? Where are you at?
So at the moment, the biomechanics approaches exist, and I developed the AI approaches during my master's.
Right.
And so we've already integrated some mechanical constraints into the architecture design. So we have like conservation of mass, but we still need the biomechanics for some of the other components, like the tissue, the stress-strain relationship of the tissue, and some of the boundary constraints.
Cool. Yeah. Good. Thank you.
Yeah.
One more quick question.
Just to follow up about the AI model, so can you briefly tell me what the training data is and what is the objective function you're optimizing during training?
So it's an unsupervised approach, and so the neural network just takes the image pairs and estimates the deformation and then resamples one of the images, moves it to match the other image. So the loss function is an image similarity between those two images, and you also have a regularizer. And so that regularizer is usually pretty simple, like just the norm of a gradient. And we want to use like the conservation of momentum equation for the regularizer. So it'll will integrate biomechanics in that way, in the regularizer. Yeah.
Thank you.
Cool.
Thank you, Matthew.
Cool.
Our next presenter is Max Dang Vu from the University of Auckland, Breaking the Wall on Breast Cancer Visualization.
Oh, thank you. Breast cancer affects many women around the world globally. In New Zealand, this is the case for 1 out of every 9 women. The best course of treatment is to locate cancers early and accurately. However, studies have shown that about 19 to 22% of patients end up having to undergo reoperations due to incomplete tumor removal. So let's explore why breast cancer localization is challenging. Firstly, a patient is diagnosed in the face-down position via imaging, but they are treated in the face-up position without any images acquired clinically. As you can see, there are huge changes to the shape of the breast and also to the location of tumors inside it. Secondly, the clinicians must use the face-down image to locate tumors, but these images are visualized on a 2D interface, which significantly limits the clinician's ability to accurately pinpoint where these tumors are. So this creates a wall where breast cancer visualization does not provide us with real-time tumor locations during the clinical procedure. The question that I have is, what can we do about that? I propose to break this wall using a physics-driven augmented reality platform. So let's break this down. I know this sounds like a lot, but here we go. Firstly, I simulate how these tumors move from the face-down to face-up position using the laws of physics. The initial simulations that I solved took a few minutes to work. However, we want this information to be provided in real time. I therefore use machine learning to speed these models up so that they could be able to solve for tumor predicted locations within a second. Now here is the interesting part. I then augment these simulations directly onto the patient using a technology called augmented reality. What I mean by this is I incorporate the simulations into some software, then upload that software onto an augmented reality headset, as you can see on the image, and that clinician uses that headset during the procedure. As they look down onto the patient, they're able to see the tumor locations visualized directly on the patient. They then take a marker, mark where that tumor is on the patient's skin surface, and remove accordingly. What I'm planning to do next is to work with clinicians and develop a study that essentially tries to understand, well, how well does my platform actually help them to accurately and efficiently locate breast cancers? If successful, this will be the first platform that leverages physics, augmented reality, and machine learning to show breast cancers in real time and be able to break that wall. Thank you.
Thank you, Max. Any questions?
Thank you. So, yeah, clearly that's a very challenging problem. So my question is probably a naive one. How hard is it to solve this problem from the other end? So basically to take the MRI image in the same position as the surgery is performed.
That's a very good
something that is fundamentally impossible, or?
So that's a very good question. So the MRIs has been taken in the same position as surgery, but one of the main constraints is that the images that we get back are very low resolution. In other words, they're not very clear, so you cannot actually see where those tumors are located. And there's also a lot of expenses that comes on top of being able to have a setup that can do that sort of imaging. So that isn't usually acquired clinically. However, it has been done in research before.
Yeah. Thank you.
Just a very quick follow-up on that. The VR set on the surgeon, would you envisage them just marking the tumour with the VR or actually doing the surgery?
I would say it's a bit of both. So the way that I've termed it is that it's for clinical procedures. So it could be for locating tumours and also doing the surgery, or like, say, like a biopsy, for instance.
Thank you, Max.
Thank you.
Our next presenter is Morehu McDonald from Te Wānanga o Aotearoa, Breaking the Wall of Genocide and Colonialism.
Tēnā koutou katoa. So, European genocide and colonialism of indigenous peoples throughout the world is a crime against humanity. The term genocide was conceived by Ralph Lemkin, a Jewish lawyer, in 1944 to address the crimes crimes against humanity by Nazi Germany. But the perpetrators of genocide against indigenous peoples for more than 600 years have not been brought to justice, nor have they received any apology for the wrongdoings. European genocide was created by the imperialistic empires of Great Britain, France, Germany, Spain, Portugal, Netherlands, and America. Under the guise of scientific discovery in the name of God, gold, and glory, without provocation, they violently invaded the countries of indigenous peoples. They stole their lands, destroyed their language, culture, and beliefs, and enslaved and exterminated the native populations. Millions of indigenous peoples around the world were killed in the process. Nations such as Great Britain and other perpetrator nations expanded across the world, acquiring immense wealth that benefited them economically and politically. For Māori, genocide started in 1769 with James Cook. By the 1890s, Māori were labelled a dying race. The population dropped from 200,000 to 40,000, and Māori land ownership dropped from 27 million hectares to 1.5 million hectares. For Māori, genocide has been like other indigenous peoples have faced around the world, which we've had to endure punitive assimilation policies, oppression and racism from white-dominated colonial governments. This has resulted in intergenerational trauma and inequities of high imprisonment, poor health, social decline, suicide, and failure in education, and forced removal of children and poverty. Breaking the wall of European genocide requires breaking down the walls of silence, apathy, and injustice by the European perpetrator nations. I've just lost that. So how can this be achieved? By the European perpetrator nations joining with the likes of the United Nations and central figures like King Charles to actually address the issues of supporting 12 indigenous countries that have put in a joint statement to call for reparations to be returned to the First Nations of indigenous peoples that suffered at the hand of European colonialism and genocide. Tēnā koutou katoa.
Kia ora, Morehu. Any questions?
Kia ora. Thank you for that. What would be the next step in terms of breaking down the walls of colonialism?
Well, I think it's the requirement on the, as I said, the European perpetrator nations to take responsibility for the wrongdoings and injustice that they have been responsible for in their colonial past, and for them also, as I said, to actually join with the United Nations and central figures like King Charles, Pope Francis, to call on a renunciation of the doctrine of discovery, and also to actually set about immediately putting in place reparations for the First Nations people that suffered at the hand of genocide and colonialism. I hope that answers the question.
Any other questions?
You've sort of answered what I was going to ask. My question would have been more about domestic processes like treaty settlements, how they fit into this much more international and indigenous nations coming together process.
Yeah, I think the treaty settlement process doesn't answer the full, doesn't provide the full reason as to why the current inequities exist for Indigenous peoples, because they're the same for Indigenous people as you would have seen in the slides there. The same historical trauma, the same inequities that they suffer, we suffer as Indigenous peoples today. So that means that the word that we use, colonialism, doesn't convey the full impact of what was suffered by the Indigenous peoples around the world. And that is the word, the proper word that we should be using is genocide, because that was what happened with the invasion, unprovoked invasion of the Indigenous countries by European nations that I named. And they are the European perpetrator nations that need to hold themselves accountable and also to provide actions, as I said, to the United Nations and jointly to solve that problem which they created against Indigenous peoples around the world.
Thank you, Morehu.
Kia ora. Kia ora tātou.
Our next presenter is Reeha Sharma from the University of the South Pacific, who is breaking the wall of plastic-based pollution.
Plastics, with no doubt, is a wonderful material which is used daily for many purposes. More than 400 million tons of plastics are produced every year, out of which about 14 million tons ends up immensely polluting our environment. When we look at this picture, we can see that the boy is eating an apple. After eating, he has thrown away the plastic bag. Now, since that bag is non-biodegradable, it will act as a pollutant and end up either in the landfills or oceans, harming the environment. We all know that majority of the plastic pollution comes from food industries. So, to break the wall of this issue, we have created an innovative type of packaging known as biodegradable edible food coating. We have used all the natural and biodegradable polymers such as cassava starch and agar obtained from red algae. So starch and agar solution were prepared and the desired fruit was dipped inside the solution, and after drying it formed a coating on top of the fruit. Now we all know with food comes many possibilities of spoilage caused by foodborne bacteria and fungi. Therefore, in our lab, we have used clove oil, which is an antibacterial and antifungal agent to inhibit the growth of microorganisms, making the film a safer environment for food. Now if you see our checklist, since all the ingredients used in this recipe are non-toxic and edible, so it can become an edible food coating. So on the days of hurry, foods can be consumed without unwrapping it. It is biodegradable, it is antimicrobial and reduces plastic pollution, such as if it's thrown away in the environment, the nature will use it up, or it can become a food source for animal. For future perspective, if this research becomes a success, it can also enhance or increase the lifespan of food, contributing towards food security. So in these few minutes, we have seen the power of nature that can be used to reduce plastic pollution by creating a biodegradable, edible food coating. So let's stop pollution and be part of the solution. Thank you.
Super interesting. What happens with heat or cold? Because obviously, fruit, for example, when it's transported somewhere is either cooled or it sits in a hot container. How does that treatment react?
So the starch and agar solution that we have prepared is like a coating on top of the fruit. So we can adjust the concentration of that starch and agar so that it can give that environment of the fruit to prevent water loss in humid environments. We know one type of preservation is refrigeration. So some places where we can't refrigerate, that coating will prevent from that particular fruit to dehydrate and at least retain moisture to keep the freshness of the fruit.
Have you done any consumer attitude surveys yet?
Not at the moment. My research is still in progress. So at this stage, we are still in the process of monitoring the freshness. And once it becomes commercialized, then from there we will try and take that consumer, as you said, yes, consumer considerations.
Thank you, Reeha.
Thank you.
Our next presenter is Robyn May from the University of Auckland, breaking the wall of newborn digital twins.
Kia ora koutou. I'd like to begin with a rhetorical question for all of the parents in the audience. Was there any greater moment of joy in your life than when you welcomed your newborn baby into this world? Well, for 1 in 10 parents, this moment is not what they were expecting because their baby is born preterm, or before 37 completed weeks of pregnancy. And in addition to the surprise, this can be quite a stressful situation because all the systems in these babies' bodies are immature, which means they run into a range of problems requiring specialized care in the neonatal intensive care unit. But even when they grow up as adults, they also have an increased risk of cardiovascular disease later in life. This is the problem we are tackling with my PhD project by developing newborn digital twins. Now I know what you're thinking, what is that? A digital twin is a computational model. That means it's a set of mathematical equations that lets us run computer simulations, in this case of how the heart beats and how blood flows through the bodies of these newborn babies. In my PhD project, as a former clinician, I went back into the hospital and I collected ultrasound scans of the heart and blood vessels of term and preterm babies, looking at both the structure and the function, shortly after birth and at 3 to 6 weeks of age. And then for my PhD in bioengineering, I developed the computational model of the cardiovascular system for newborn babies. Finally, we put it all together as newborn digital twins. So for each and every baby that we have ultrasound data for, we now also have a personalized heart model for that baby. This represents one of the largest cohorts of human digital twins and the first in newborns. I believe that these newborn digital twins are going to break down walls in clinical care because they can provide personalized predictions of blood pressure and flow that can help clinicians make those difficult decisions in the neonatal intensive care unit. And I also believe that newborn digital twins can break down walls in physiology research. By comparing term and preterm digital twins, we can get a deeper insight into what's different in babies who are born preterm. And that's the first step towards future research, to being able to reduce their increased risk of cardiovascular disease later in life. Kia ora.
Any questions?
Thank you very much. Very interesting talk. So I can imagine that modeling this motion dynamics of the heart and blood fluid motion and all those very complex processes is very non-trivial. And as we've seen recently, AI models can often be really, really good at those things. So what do you think about that? So would there be enough data? Would they be helpful in the future to solve this problem?
Yeah, I think, I don't work in AI, but I think you need a lot of data to train those models. And as someone who just tried to collect ultrasound data, it's not easy to collect high quality data in newborn babies. So what we have here is a biological model that's also physics-based, which means we can be sure that what we're, the predictions that we're getting are actually biological plausible.
And when you're making those predictions about what will happen later in the lives of those newborns, what data are you basing it on? If I understood correctly, the data that you have is from newborns who are now nearly newborns, I guess. So you don't really have the data from their future.
Yeah, absolutely. So we collected data shortly after birth and at 3 to 6 weeks of age. And you wouldn't think that such a short time span is enough to see a difference. In our clinical data, we didn't see any differences between the term and the preterm groups. So if we relied on that alone, we'd say there's no evidence of cardiovascular remodeling. But when we augmented it with our modeling, there were actually differences that the models could show that we would have otherwise missed. For example, by term equivalent age, preterm hearts are actually stiffer than term babies, which could contribute to hypertension somewhere down the line, increased risk of heart failure, and that's the mechanism of that increased risk of cardiovascular disease later in life.
Thank you.
Thank you, Robyn. Our next presenter is Savindi Wijenayaka from the University of Auckland, Breaking the Wall of Gut Microstructure.
Do you know that 3 out of 10 in the world suffers from a digestive disease? The statistics are same for New Zealand, and Maori and Pacifica population is one of the most affected. For some Kiwis, for example, the 15-year-old Eva or late Ruby, it's more deadly and also very painful. The image next to it shows a 5-year-old Thomas's drawing of nausea monster, explaining his feelings towards nausea. So as you can see, it's not a pleasant thing. So their stories highlight our limited ability to accurately diagnose and treat these conditions. We now know that the sphincters, those muscular valves that control the food movement in and out of the stomach, could be a major reason behind these diseases. However, one of the things that make the sphincters so tricky to understand is their structure. Even the most detailed image ever only shows a flat, two-dimensional cross-sectional view, when in fact, story is a lot more complex. So here's how I tackle this challenge in 3 key steps. First off, I collected rat stomachs post-mortem, and then I used contrast agents to highlight different muscle layers, and also used micro-CT to get the detailed images. Finally, with the help of supervised machine learning, I labeled the different muscle layers and reconstructed the 3D structures. I also calculated different muscle properties like the muscle thickness and fiber orientation so that we can get a proper idea about these things. So one interesting finding so far is the unique muscle structure and also the uneven thickness around the sphincters, which showcases a radial asymmetry. To understand the importance or significance of these findings, we will incorporate these into computer simulations and also validate it with the Fraunhofer Institute. With these simulations, we will be able to understand how the stomach functions are affected when the sphincter structure is deteriorated due to a disease. So this way, we can help patients like 15-year-old Eva by breaking down our understanding of gut sphincters. Thank you.
Yeah, I'll go first. So can you elaborate a little bit more on the very last sentence that you said? So how exactly are you envisioning helping those patients with this technology?
So, like I mentioned, one thing is about the sphincter structure, that we don't know how that affect in the different kind of diseases, and also we don't know the exact muscle architectures of the sphincters. So we try to understand the basics first so that we can build up on that, for that, you know, we can make cool things out of it. That's the main idea behind my research. So we get the basics straight, so we have more knowledge on how the sphincters are deteriorated and how that deterioration affect the stomach functionality. For example, if we take gastroparesis, how the sphincter structure contribute to that aspect. If the sphincter is, let's say, thin, would the push is more, something like that.
Thank you. One more question or not?
Yeah, just that brings me back to the sort of first point you made about identifying where the cause lies, and I wondered whether you've looked at, say, somebody being temporarily fed intravenously, whether that solves the problem of nausea, whether it is really in that transition of food entering the stomach.
Could you repeat the question again?
Yeah, just whether you've looked at to identify the cause in the first place, whether how confident you are to be able to say it is at that point of transition where the food enters the stomach.
Yeah, so when the sphincter is not working properly, the closing doesn't happen properly. So then you get the food coming into the esophagus, so that would cause nausea. Yeah. So that's why the sphincter is so important. Yeah.
Thank you, Savindi.
Sorry.
Our next presenter is Shallu Verma from University of Canterbury, who is breaking the wall of illicit drug analysis.
Thank you.
Imagine a world where mathematics becomes our ally in solving the most arithmetic conundrum, quantitative analysis of drugs. So in New Zealand, almost half of the population has tried drugs once in their lifetime, and methamphetamine alone has caused $800 million harm. And overdose deaths, which is increasing every year. Now, why this puzzle eludes an easy answer? If you have a look at these packets, they look clearly similar, but they are not. So can you identify them? And when you buy drugs from the drug market, which is like the authenticity is not at all clear, so how can you trust on them? Now, if you look at this paracetamol, if you have a fever, would you take 1 or 10? Obviously 1, otherwise a trip to hospital awaits. So how can you tell the dosage in these packets where the people are going to sniff or try? So quantitative analysis these days happen with these 4-step journey, and ideally it takes about 4 hours. And just like ideal gases, ideal situations do not exist. And they also come with further drawbacks, which is they are very expensive, requires reference standard and calibration curves. So what if we could change the whole game? In my project, I am using NMR, not high field one, but the benchtop NMR. It is compact, easy to use, and cost friendly. Now, why this is different from high field? It doesn't use cryogens. It does not need a big space, just like it's small like a printer, which can be fitted on the bench top. Now, we do not stop here. We are using our unique model, quantum mechanical model, which is field invariant and which can resolve the peaks quickly in the mixtures like these and help in analyzing the drugs in just 20 minutes and making the quantitative analysis quite simpler. The major impact of these projects is on harm reduction centers and forensic labs, which can help the healthcare. Thank you.
Thank you, Shallu.
Thank you.
Any questions from the jury?
Very interesting talk, thank you. So I guess this all sounds very impressive and like a major advance in the technology, but I'm guessing it should be coming at some cost. So what is the cost that is incurred by reducing the size and making the test available out in the field?
The cost of high field NMR is about $1 million to $3 million, but the benchtop is about $50,000 starting from.
Right, but does that reduce the accuracy or is it?
Yeah. As the high field magnets have 500 megahertz about, and its frequency is 60 megahertz, so it reduces the resolution, which makes the analysis quite time consuming. But to reduce the time, we are using this quantum mechanical model software, which can reduce the time quite effectively, and it is field invariant, so we can even have the spectra of high field and load into that model, and we can analyze the peaks thoroughly. To make it more simpler, we are trying to load the prevalent drugs like methamphetamines, cannabis, like that in the model automatically. So as the drug come or case come, we just can load and see the peaks and quantify directly.
If you have mixtures of drugs,
yeah.
If you have mixtures of drugs, how does that, will it still work fine?
Yeah, as I showed in the figure, like I showed the example of caffeine, acetone, and ethanol, and it was able to identify the separate peaks from all of the molecules.
Cool. Okay.
Yeah.
Neat. Thank you.
Thank you.
Thank you, Shallu. Our next presenter is Sherry Feng from Auckland University of Technology, who is breaking the wall of bias in AI.
Imagine a supercomputer that can do anything that you teach it to do. Sounds amazing. What could possibly go wrong? Let's say you want the supercomputer to be able to screen resumes for a particular job. And let's say let's say you want the supercomputer to be able to screen resumes for a particular job. You'll need to provide it historic information of successful and unsuccessful applicants. If this was for the tech industry today, which is male-dominated, your successful applicants tend to be males. You can see where this might be going. So once our supercomputer sees a resume as a female, it's more likely to reject it. Why? Well, we just taught it that successful applicants tend to be males. What I just described to you is a significant issue that is happening in our AI systems and the consequences of having implicit bias. The bias comes from information and data from our real world, which is then fed into our AI systems. While we're still in the process of eliminating prejudice and bias in our society, our existing AI systems, along with the information they process, already has such bias in it. So what can we do? Well, since we can teach our AI systems anything, can we teach it to not be biased? How? So let's take, for instance, an individual who may have a bias or stereotype that all girls wear skirts, because all they've seen are girls wearing skirts. One way to eliminate this stereotype is to show them examples of girls wearing pants. And given that our AI system is built to mimic the human mind, we have been trying the same method. So integrating new data into our AI systems, like examples of girls in pants. But because of the existing data, the AI system actually no longer recognizes them as girls. So in my current research, I have developed models and algorithm that is able to take such new data and be able to integrate it into the AI system while still without changing the original features. And this extends beyond gender, encompassing various features and traits of an individual that may induce implicit bias. Ultimately, my research vision is to make all AI systems fair, especially if they're helping to make decisions for our world. And my research starts by breaking down the wall of bias in AI. Thank you.
Thank you, Sherry. Any questions?
So yeah, I'm trying to understand how you identify those biases. So I can imagine coming up with a list of known biases that we are all aware of, and then training your model to avoid those. But what about the unknown, unconscious biases in the sense that, you know, we train a model, it is biased to something, we don't know what it's biased towards. So can we adapt the model in a way that it will be identifying those hidden biases and fixing them?
So you're talking about in terms of identifying it first before we reduce the bias?
So I'm just guessing that you will be giving a list of biases to the model that then will be fixing.
Yeah, so basically it's going to just solve any systems that already have bias, and you'll probably see it just from an output. You'll find that, yeah, for instance, the example I gave with the resume, if it tends towards males, then it's obviously pretty biased. Another way is also looking at the features that it takes. So when you feed into AI models, you can see which features it will take. For instance, like, you know, for an individual, if you have an image, right, you take features such as the clothing, you know, like size of the person, or even like how they look. Whereas and you can also see if, you know, maybe gender or skin color becomes a feature or a factor that it takes in to make its decision. So yeah, that's, I guess, sort of two ways to see the bias.
Thank you.
Yeah, thank you.
Any further questions? No. Thank you, Sherry.
Cool. Thank you.
Our next presenter is Tejesvi Patel from the University of the South Pacific, who is breaking the wall of infection using PVP-TA.
Hello everyone. Picture this scenario. You're having a lovely day when in a moment of clumsiness you hurt yourself. That wound is now an ideal environment for bacteria and fungi to grow and cause infection. Due to rising global temperatures and the constant misuse of medication, there's been a substantial increase in the number of microbial infections, especially in terms of skin infections. Microbial buildup on wound sites impedes the natural healing process and slows down healing. There are common solutions to this in terms of ointments and tablets. However, these have drawbacks of their own. Ointments require constant reapplication due to being washed off or they're being diluted. Tablets are not site-specific. They are slow-acting. They have substantial side effects, all because the body has to metabolize the tablet, and then the medication is carried throughout the whole body and not just one infected site. My solution to this problem is the use of PVP-TA hydrogels that have been loaded with an antifungal drug, nystatin. Now, PVP is polyvinylpyridone. It's a synthetic, biodegradable, biocompatible polymer that's used in a lot of everyday products, such as contact lenses or capsule coatings. Tannic acid is a natural polyphenol that's found in plant material, such as oat bark and tea leaves. Our research has shown that when these hydrogels are placed in fluid, both the loaded antifungal drug as well as tannic acid release. Now, tannic acid has a lot of useful functions. It's antibacterial, anti-inflammatory, it stops bleeding, and current research has started looking into its anti-cancer properties as well. Thus, the use of this hydrogel in the form of a topical patch would present a material that is site-specific, is fast-acting, would have substantially less side effects, and would not require constant reapplication, all because the topical patch is applied directly to the wound site. But most importantly, the topical patch should be both antibacterial and antifungal due to the release of both nystatin and tannic acid. Additionally, the topical patch would also be completely biodegradable, thus alleviating the plastic waste that's commonly associated with conventional treatment methods. These topical patches would especially be helpful to diabetic patients who are more prone to infection due to slowed wound healing. Thus, we believe that through the use of PVP-TA hydrogels that have been drug-loaded, we can greatly assist break down the wall of microbial infection. Thank you.
Thank you very much for this. Can you tell me a bit more about tannic acid? Is it used in other ointments if it has all those properties?
So tannic acid has been used back in the days for like burn treatments. It's currently used in things like leather tanning now. It's not as commonly used in medication nowadays, but it's making a comeback slowly in terms of cancer treatment or even just wound healing in general. But it's not as commonly used as it should be.
Have you got to the point of clinical trials or anything like that? Where are you at?
So unfortunately at our university, we cannot go into the stage. So that's the wall that I'm currently facing. But we have done things like in vitro tests on like petri dishes with bacteria and fungus, and we've seen pretty good results.
Thank you.
Thank you, Tejesvi.
Thank you.
And the very end, the next presentation is Vitor Ginnicelli da Silva from Ritter Institute at Massey University, Breaking the Wall of the Infant Microbiome.
Digestive diseases affect 443 million people globally, a number that has increased by 74% in the last 30 years. Just in 2019, 89 million years of health life were lost due to these diseases. But they are not just numbers. They are people like me, you, our loved ones. We need the urgency to solve this challenge, and one of the answers may be in our guts. Gut microbes strongly influence our health. In turn, they are mainly shaped by our diet. Good nutrition during infancy is indispensable to avoid imbalances in the gut microbiome, which increases the risk of diseases later in life. However, traditional laboratory techniques provide a limited understanding of the interaction between host diet and gut microbes. At the Ritter Institute, we are breaking the wall of the infant microbiome by applying mathematical models to investigate a decisive but understudied point of life: weaning. We used the DNA of gut microbes from weaning infants to represent their metabolism as mathematical equations. Then, we employed the computer codes to solve these equations. This allowed us to quickly and inexpensively predict how gut microbes react to a diet. Using this strategy, we screened a wide variety of foods consumed by infants in New Zealand. And we identified the candidates that increased the most the microbial production of beneficial molecules. These foods may help us to keep a balanced microbiome from early on, contributing to the prevention of digestive diseases. But the application of mathematical models can go far beyond this project. For instance, models can be used to differentiate the microbiome of healthy and sick individuals, or to predict how gut microbes metabolize drugs. Models have the potential to largely accelerate the study of microbial communities. In the end, this knowledge could pave the way for revolutionary advancements in human health. Thank you.
Any questions for Vitor?
I'll go with one. Thank you very much. In those weaning infants, have you got infants both breastfed and bottle fed?
Yeah, so the data that I used, there are 14 infants from New Zealand and both mixed the types of feeding. And they were aged between 6 to 12 months old.
How much variation do you find across New Zealand? You've got lots of case studies? I imagine lots of samples and
actually, no. That's a point that I'm a little bit disappointed. Honestly, there is not much investigation. I mean, there is investigation in New Zealand, but not the type of data that I can use to fit my model. So that one, unfortunately, it was only 14 infants. That was the best that I could do.
Right. Is 14 going to be enough?
I mean, what I would say, in the end of the day, the whole idea of these models, when they get better and better, is to create the personalized nutrition. That's the main aim. So ideally, even one in the end would be enough.
Yep. Okay. Thank you.
Sorry, just one more question, Nate. Do you expect that there'll be differences between different ethnic infants from Māori and Pacific and I don't know what the ethnicity of these 14 infants
So that's a very good question. You know that the gut microbiome is quite a personalized thing, and ethnicity is one of the factors that shapes, and it's very related to the type of food, because of course, if you come from different cultural backgrounds, we eat different foods. So yes, I would say probably there is, but there is a huge problem also on the studies of the microbiome because indigenous populations have been neglected, and even to do the simulation of this type of foods, like I use the American databases, of course they don't have a native New Zealand food, so there is this limitation as well.
Thank you, Vito. So that concludes all our presentations from the selected Falling Wall contestants. Fantastic talks. Please join me in thanking them all for this. It's been really, really exciting. Right, so what happens now is that we now leave it up to the jury to find the difficult task of selecting a winner to represent New Zealand at the Falling Walls final in Berlin. That will take about an hour or so. In the meantime, we will have a couple of talks here now, and then there will be some video showings from a video series we did about the Whittingham Fellow awardees. So I'll just start out by introducing Tia Haira. Is Tia here? So Tia is a representative for the Royal Society Aparangi's Early Career Researcher Forum. And since many of you are early career researchers, or all of you are, we thought it would be a really good idea to give Tia an opportunity to just talk about the forum and what the forum does. So thank you for coming today, Tia. Oh, I should just say, the jury, you come with me, and I hand this over to Tia. Are you mic'd up, or you use this?
I think I'm just going to use that.
Thank you.
Tēnā koutou katoa, whaia te tikahirangi ki te tū ohu koe me he maunga teitei. Ko te mea tuatahi ki te kaiwhakahaere o tēnei hui, o tēnei kaupapa, anei te mihi kia koutou. Ki ngā kai kōrero, anei te mihi kia koutou. Mō tō koutou mahi, I o rangahau, I o mātauranga, he tēnei miharo ngā kōrero. Kia koutou, kua whakamene mai nei ki tēnei huihuinga, ki tēnei kaupapa, ki o koutou kai ngākau, ki te aro mai, ki te awhina I tēnei kaupapa. Nō reira, tēnā koutou. Ko wai ahau, ko te aroa waka, ko te aroa tangata. Ki te tau tāku māma, ko tuarangi Ngāti Wāhiao, ko Ngāti Whakaue, ko Ngāti Rangi Te Arere, ngā iwi. Ki te tau tāku pāpa, ko tūwhare toa, me Ngāti Rangi, me Ngāti Pikiawa hau. Ko Tia Haira tāku ingoa. Nō reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. So my name is Tia Haira, and I'm a member of the Early Career Researchers Forum Committee, which is hosted here under the Royal Society Te Aoparangi. And I wanted to start off by first thanking our organisers, the Royal Society, for putting on this amazing kaupapa, and to truly highlight some of the research of talented New Zealanders. Oh, aroha mai. Secondly, I wanted to also congratulate the speakers that were selected to present here today. I've been amazed by all your work and listening to your research, and I wish you all the best for the competition and your research moving forward. To everyone else that is here to support this kaupapa, it's amazing to see the support. Nei te mihi ki a koutou. Today I'll be talking to you about the Early Career Research Forum Committee, which is pictured here in this photo, and I will explain a bit about what we do and who we are. So the Early Career Research Forum Committee is a network of early career researchers hosted by the Royal Society Te Aoparangi. And as a forum, the forum is spread across, forum members, are spread across the research and science sector and come from various organisations, disciplines and locations. The forum is dedicated to engaging New Zealand early career researchers, it's very sensitive, early career researchers on issues that are important to them, and fostering a collaborative, informative and respectful community. Our forum seeks to represent a national voice for New Zealand early career researchers and to celebrate their achievements and their contributions to their respective fields. The priorities of our forum seek to address issues around equity and diversity, strengthening our representation of Māori and Pacific researchers and Māori and Pacific research methodologies. We consider the current research, science and innovation environment and how we can better support early career researchers. The Royal Society supports our forum to organise events, publications, advocates for our aspirations on political and research platforms. We hope to continue this work, discover new and better ways of doing things, and paving the way for our future generation of researchers. So earlier this year, we had a 3-day wānanga called He Pito Mata, which provides early career researchers an opportunity to make new connections and debate issues such as employment, stability, precarity, funding, and support. Oh my goodness, sorry. In particular, ECRs discuss and draw attention to what conditions allow ECRs to thrive, and possible ways to rethink the current research, science and innovation sector to address both their hopes and concerns. A strong vision of early career researchers is to see a sector founded on strong relationships, more stable employment, and seeks to deal with inequities. This conference is a great way to network with each other, so if you haven't already heard about it, I would encourage you all to attend the next conference. The name He Pito Mata and the kaupapa is taken from a whakatauki, Iti noa he Pito Mata, which refers to a small uncooked portion of kumara that was replanted to produce many more kumara. In the ECR space, it refers to awakening the potential of early career researchers in Aotearoa. As a forum, we take the learnings from He Pito Mata to provide solutions to combat issues facing ECRs. ECR is a self-identified term, but usually refers to someone studying towards a master's or PhD, or having completed, or up to 10 years, post-studies. All ECRs are eligible to sign up to the Royal Society Siaparangi newsletter, or as a professional member. You can also sign up for the newsletter, which also keeps you up to date with what's going on and other opportunities such as funding. And more information can be found on the website. There's also opportunity to join the Early Career Research Forum Committee, who work closely with the Royal Society. This allows us access to key stakeholders and advocate for better conditions for ECRs, as well as organising events such as Hipi Tomata and workshops to help further develop skills and competencies of early career researchers. Please consider joining the Royal Society ECR Forum mailing list and the Royal Society as a professional member. And if you have any interest in being involved in the committee, please email us or speak to me afterwards. We have rolling vacancies of positions replacing outgoing members. With that, I'd like to leave you with this whakatauaki by Tahimi Hinarere. Kua tawhiti ke tō haerenga mai, kia kore e haere tonu. He nui rawa o mahi, kia kore e mahi tonu. You have come so far not to go further. You have done so much not to do more. Thank you all for your time this evening, and once again, good luck to all our speakers. Mauri ora koutou.
Kia ora. Thank you so much, Tia. Kia ora. Tēnā koutou katoa. Thank you all. What an inspirational session. Hearing about all the hard work and determination that's gone into your research and how you're able to translate some of that curiosity and passion for the rest of us is exactly why we come to work every day at the Society. So my name's Tara Satyanand, and my team works on how the Society makes connections. So we have nearly 1,500 members, and of those, some incredible scientists, researchers. There are a set of elected fellows and companions, and also 40 constituent organisations. Those are science-related associations and societies, and also affiliated organisations. So it's an incredible resource, this network that we can tap into, and I completely agree with Tia in encouraging you to join. If you're not already a member, please at least sign up for our alert newsletter or follow us on Twitter or Facebook or wherever you are on the socials, because there's a huge range of opportunities, funding, networks, mentoring that we'd love you to be able to tap into, and we'd love to stay in contact with everybody who's presented here today and support you along your career journey by linking you with other people across our network. Thank you also to the German Embassy for your support for this kaupapa with Falling Walls, and we're lucky enough to be able to tap into experts like we have on our jury today, who have the difficult task of choosing which of those presentations, will be the person who will be able to go and represent Aotearoa New Zealand or Fiji internationally. So thank you to Tia, who's part of our Early Career Researcher Forum. Next, I'd like to introduce Jules House. So Jules is here from an organisation called Momentum, and this is an organisation that has been set up, again, to create links for young researchers and to push research along the pathway towards creating impact in the real world. So Jules has a background in commerce and law, based in Christchurch. He's linked with Return on Science, which is one of the commercialisation organisations based out of University of Auckland. And I'll pass over to Jules to talk to you about how his organisation invests in exciting research.
Hello everyone, and thank you for coming today, and congratulations to all the speakers. I know that I'm the last speaker and you probably don't want to hear anyone talk anymore, but if I can get maybe 5 to 7 minutes of your time, that would be really, really appreciated. And moin an der deutschen Botschaft. Schön, euch alle heute kennenzulernen. Ich hoffe, wir können später vielleicht sprechen. So yeah, I'm with Momentum and Return on Science, and they are part of the Commercialization Partner Network. So what is Momentum? It is a series of 5 investment committees that are based all around the country, and we are here to provide researchers, students, or faculty staff members and startups the best chance at commercial success by connecting you guys with people in the industry and experts who can help you scale up your current research into the best commercialization strategy possible. So yeah, we aim to increase the quality and quantity of the commercial opportunities that come out of New Zealand and really try to make New Zealand a global player in the startup world. And I think we're doing a good job at the moment. It just takes everyone to kind of play along as well and actually realize the potential we have in this room. And how can Momentum help you? So if you have an idea, you can come present to an investment committee and ask for recommendations, advice, or access to funding. And you might ask, so what does an idea mean? How far along do I have to be? Well, we've got 4 criteria on how to be involved, and you only have to meet one of them. And it's any idea, at any stage, at any time, at any place. So you can be in the shower in the morning, come up with an idea, and if there's a slot in the evening, you can come present to the investment committee and just talk to the committee. You don't have to have a presentation. So sometimes what we've had in the past is that the first project might be a startup that's just raised $3 million in seed funding, but they don't know what to do with the next steps, or they're currently just talking over the next steps to get some approval. And the project after that will say, last week I rolled out of bed and this happened, and now I'm trying to fix it. What do you guys think I should do next? So we see a very wide variety of projects, and yeah, so don't be afraid that your project isn't far enough. We actually encourage you to come earlier so we can answer those tricky questions that you might have at the start of a project. And it is a very safe space to share your idea. It isn't like the Dragon's Den or Lion's Nest or whatever you guys call it. We see the investment committee as an extension of your team for that 1 hour. So yeah, use it as best you can. And the way it works is that you present for 10 minutes. After that, it's around a 30 to 40 minute Q&A session where the investment committee will ask you questions similar to what the juries did today, and you get to answer. And we really encourage to answer with, I don't know, if you don't know a question as well, since that's the best way we can help you too. Yeah, so we got world-class advice. We've got industry experts from all around the world. They zoom in or fly in as well to just come and sit in for the day to listen to your project. One of our investment committee members has the personal phone number of Bill Gates, Jeff Bezos, and the founder of Starbucks because they used to work together. And those are kind of the connections that our alumni has. Other people have built parts on the International Space Station, and now they have an app on their phone to make sure that the space station isn't over their head because they're not confident with what they built. And yeah, we've got amazing resources as well to help you on your next steps, no matter what stage you really are at. As mentioned earlier, we've got 5 investment committees, and they're all over New Zealand. We've got one in Auckland, Canterbury, Manawatu, Otago, and Wellington. But if you're in Wellington and want to present to the people in Auckland because you're seeking a certain expertise, you're more than welcome to come join via Zoom, or if you're in Auckland on the day, you can do that as well. We meet up every second month, and it's free of charge if you are associated with any kind of tertiary institution, which I assume everyone in this room will be. And if not, we can work something out to make it free of charge as well. And you can come back as many times as well. Sometimes projects will think they can only come and present once. But again, as mentioned earlier, we encourage people to come as many times as they can and as early as they can. The only condition is that we ask them to come back with progress, so that when they come and present again, it's not that we say, have you worked through this? And like we said last time, and they say no. So we just recommend to work through the last recommendation set before you come back to present. And how do you get involved with Momentum, you might ask? It's very, very easy. you can either go on our website, momentum.ac.nz, and there's a box in the top right or in the bottom left, that white box. The other option is to either send me a LinkedIn message or an email and just say, hey, Jules, I would like to present. Can you please tell me what the next available time slot is? And that's it. That's all we need to do. And we'll probably fit you in. We don't want to make anything a barrier for you to come and present. We want to make the application as easy as possible and the journey as smooth as possible. And we're happy to ask any questions too. There are some forms to fill out, but they are only really to provide the investment committee members with some background information on your project so they know what kind of questions they can ask you or what kind of problems you might be facing for your next step. And this is just the next page, and it's really quite simple to fill out. There's also the other side of Momentum. It's not just presenting, but being a part of a Momentum investment committee. So we're always looking for students to join. It doesn't matter what stage you are. You can be an undergrad, you can be a postgrad, you can be doing a diploma. And it really is a great experience to be an investment committee member. You'll be one of the first people to see New Zealand startups coming through the ranks, and you'll have a direct impact on what they'll be doing next as well. We also host a lot of capability development sessions. This year we had a 3-day conference in Auckland where we flew all of the investment committees up and had speakers come and present and workshops. It was a really, really good time. We also have something called the Momentum Internship, where people can apply to some of our partner venture capitalist funds that we work with. So you can work there for 6 months to see what it's like to work at a VC. And you might think, oh, I'm not interested in that. But some of the venture partners used to be PhD students working in a completely different industry, but then they did an internship with a venture capitalist firm, and now they're one of their best employees. There's also the other side, return on science. So these are more vertical scale, specialized investment committees. So we've got agri-tech and food, biotech and pharma, digital technologies, medtech and surgery, and physical sciences. Most of these meet up once a month. Agri-tech and food and medtech and surgery meet up around every second month. However, quite often in the last few months, there's been such a demand for these investment committees that we've hosted a lot of out-of-cycle meetings. And the process to apply for return on science is the same as Momentum. Just send me an email or LinkedIn and say that you'd like to come and present and get some advice. And again, very similar to Momentum, we've got amazing expertise on the investment committee. The structure of presenting is the same as Momentum. The difference is just that return on science is much more specialized and industry focused. We can also connect you to potential funding resources, whether that be public or private funding. It just depends what's best suitable for your project, or if you're even interested in funding. That's a completely different story as it is. These are some of the projects we've seen, and there are some really, really cool names on it. Limetry is a really big name. OpenStar Technologies, you might have seen on the news. Cirrus Material. Eat Kinda is our winner from Student Entrepreneur of the Year last year, and they're making cauliflower ice cream. Kiwi Fibre Innovations are basically an alternative to carbon fibre out of harakiki fibres. And there are many, many more. Another really awesome startup that you might have seen in the news recently is called Kiki. They used to be Easy Rent, and they've just raised $6 million USD in seed round. And they came and presented to Momentum in 2020 and were about to go bankrupt. And now they've just pulled off one of the largest seed rounds in New Zealand's history, which is just incredible. In total, we've seen over 1,400 projects come to return on science and Momentum, and these are unique projects. We've approved over 20 million of investment for various startups at various stages, and see a lot of project previews. Again, like I said, you can be at any stage. You don't have to be at the stage that you're looking for investment. You can really just be there to say, is this something worth pursuing, or should I look at something else? And this is our team. Really, really awesome team, and we're spread all across the country, as you can see. So if you're in one of these cities, feel free to reach out to the person directly on LinkedIn or their email. And their email is really easy. It's just firstname.lastname@auckland.ac.nz. Or feel free to reach out to me via LinkedIn, Facebook, well, not Facebook, but email and what's the last one? Anyway, one of these. Or come talk to me in person. And there's my email if you want it. And happy to answer any questions if you've got any. And congratulations. Question in the back.
Is it always for economic kind of gain? Like are the projects always about?
No, they're not always about economic gain. We also are there just for the community gain as well. And we're not just there to measure economic gain. But it's just a bit harder to measure, I guess, community gain or cultural gain as well. But yeah, you don't have to be there just to say, like we've had charities come through before who are not-for-profit and just wanted to get advice on governance since they were kind of struggling on that direction. Yeah.
You're a lawyer.
I'm admitted to the bar, but I'm not a practicing lawyer.
My question is, you make our idea. What happens to our idea?
So we don't have any funding ourselves, and everyone in the investment committee, all the members have signed an NDA, and all the people who come and sit on so an NDA is a non-disclosure agreement. That means that everything in the room that's being said is being kept in the room, and that is to ensure that we can have an open conversation without leaking any IP, since we know that's a very integral part of any of your guys' research. And yeah, if there is an investment, it just depends on what it is like. I can't really speak on that since each situation is different. But generally speaking, momentum and return on science, we don't hold any money. We just approve funding requests from third-party funds since they're looking for independent advice on whether or not they should make an investment into a startup. Hope that answered the question. Awesome, thank you so much.
Kia ora. Thank you, Jules. So next, we'd like to present some videos. And these are because during COVID, obviously it was a really tough time for researchers around the country, and particularly for early career researchers who might have been on soft funding and didn't necessarily have that stability. So after a lot of advocacy, including from the Early Career Researcher Forum, which Tia is a part of, MB agreed to announce some funding specifically to meet this challenge, and partnered with the Royal Society Te Parangi to select 30 early career researchers. So we had $10 million to distribute between those researchers and create a series of Te Whitinga fellowships. And so there were 30 of those. We've created 10 videos which we'd love to show you today, and these are also up on our website if you'd like to have a look. So I think those should be queued up. Enjoy.
Kia ora, I'm Greer. I'm an early career researcher at GNS Science. I'm an MB Science Whitinga Fellow. We are looking at reconstructing what rainfall and wind strength was like in the last 20,000 years. So we're looking back in time. I applied for a Te Whitinga Fellowship because I just handed my PhD in February 2020, and then they went into lockdown in March 2020, and the whole entire country and the whole entire world, there was cuts in funding, there was things that were advertised were just taken away. It was really hard to get out of the country, it was really hard to get into other countries, so it just became really difficult, and it felt really unsafe to go anywhere else. If I hadn't got the fellowship, I probably would be doing precarious work or be unemployed, or I would have had to move outside of research and find employment somewhere else, or we would have had to move. So I feel very privileged to get the Whitinga and be able to continue doing research, which I really love, and continue doing it in New Zealand and in the town where we already lived. So my family was able to stay where we already were and where my husband had a job, my children were going to school. This fellowship is opening up opportunities for me to build relationships with iwi in the South Island, where a lot of my research is based. So that's really important to me, so we can create better science. This fellowship also gives me the opportunity to collaborate further with other scientists within GNS and within New Zealand and become more part of the community. One thing I really, really love about the job is doing field work. Like, the reason I started doing geology was to do field work. I get to go to really nice places, and the work is hard, and it snows and it rains and it's cold and you get saturated, but it turns out you're still having a good time. It's a great way to start interacting with the cores you're going to be using, because you can see you're in the landscape that you're going to be trying to reconstruct the climate with. So you're out there on the lake, you're out there on a peat bog, you're out there in a fjord, and you're doing all those things, and you're there with your colleagues, and it's just a great way to be together and be part of a team.
Kia ora, my name is Remy and I'm an MB Fetengah Fellow at the Department of Microbiology and Immunology at the University of Otago in Dunedin. I'm originally from Yemen. My family helped me explore the world. So I came to New Zealand as an undergraduate back in 2009. I did my undergraduate degree in biomedical science in infection and immunity because I'm just in love with the science of biology. And then I continued my honors, and then I did my PhD. And that's exactly when the civil war broke out in Yemen, and that's exactly when I also met my wife in New Zealand. Unfortunately, I'm not able to visit my family, and this has been going on for 9 years. I wanted to introduce my wife to my family, but it was really difficult. And I cannot wait to actually visit my family anytime soon. So after my PhD, I met Professor Bruce Russell, and then he said, okay, I don't want to work with only malaria, I want to work with Cryptosporidium, which is a very close relative, really hard to get rid of. Surprisingly, the cases in New Zealand are really high every year. So what we're trying to do is to develop a potent drug against that parasite. Bruce pretty much told me, all right, set up your lab, do whatever you want. He pretty much gave me green light, which was pretty scary in the beginning. And after that, we just continued on applying for more grants, which unfortunately we were not successful because it was highly competitive. And then COVID struck, which made it even harder for us because most of the research funding was directed at virology research. And we are at parasitology research, so it was really difficult. So that's when the MB Fetengah Fellowship came out, and we gave it a go. And here I am. I'm really fortunate to be one of the successful candidates, which allowed me to continue my work, and not just my work, also the work of my students. So ultimately, we want a drug that targets the parasite into every life stage that it's going through, so that we can stop transmission into other hosts. And we're going to try these compounds and see if they work against this disease, so we can effectively control cryptosporidiosis in New Zealand and hopefully the world.
Hi, I'm Febelyn Reguyal from the Department of Civil and Environmental Engineering at the University of Auckland. I'm an MV Science Fetengah Fellow. I evaluate the environmental impact of electric vehicle adoption in New Zealand using life cycle assessment, or LCA. In 2020, I attended the New Zealand University's Women in Leadership program. One of the speakers is Professor Kathleen Campbell, who is a geologist, and one of her tips that really stick to me is that always be prepared. And in 2020, I always keep in mind that when opportunity comes, I will make sure that I'm ready for it. And then in 2021, so this Fetengah Fellowship happened, I already knew what I would propose that time. And then all I have to do is just really write it, and hoping that I will get the fellowship. I will continue teaching sustainability and life cycle assessment at the university. But I got excited because I think that's an opportunity for me to finally do research again, because I think it has been like 3 or 4 years that I haven't done like a complete research. Now I have the resources, at the same time, the time to do the research for 2 years. The main part of the research is to provide baseline information to New Zealand government on how to be ready and be able to mitigate the potential environmental impacts of electric vehicle adoption. My department know that I really want to be in a career of sustainability and life cycle assessment, and they have allowed me to attend trainings and seminar in these fields to be able to actually develop myself. If you're not successful, always just try and try. One day or another, you'll be able to be successful in your proposal. And I'm sure a lot of great leaders in the research field right now, they also failed several times. Learning from all these failures would be the best way to actually succeed in the future.
My name's Paul Brown. I am a research fellow here at the University of Waikato, and I am a grateful recipient of the MB Science Whittingham Fellowship. I do some research looking into algorithmic bias, what that might look like in Aotearoa, New Zealand, looking at how we might develop frameworks for decolonising operationalised algorithms, extending that, how we might build Indigenous algorithms. The Whittingham Fellowship has opened a lot of doors in terms of being able to meet and collaborate, especially with police. I'm currently working with the Waikato District Police on the modelling of residential burglaries in Hamilton. I've also been given the opportunity to work alongside research groups in a large police project called Understanding Policing Delivery that is led by Kim Workman, who is just an amazing person to talk to when it comes to criminal justice and the system here in New Zealand. We're doing some interesting work trying to understand how police engage with communities and also how they deliver their services. I'm thankful to be leading the quantitative stream on that project, which is really exciting. It's also opened doors in terms of being able to go to conferences. I was fortunate enough to go to Italy last year to attend the International Workshop of Statistical Modelling. And that was a great conference to present my work. Got a lot of good feedback from it. That opportunity would not have been possible without the fellowship. Also grateful for the opportunity of meeting Indigenous researchers at the conference in Arizona. It's going to be really fascinating to meet other Indigenous researchers in the AI, tech and data algorithm space. I've been really thankful to be able to meet some great, amazing, super intelligent people that are also as passionate as I am about whether they be in the field of statistics, data ethics, Indigenous research. It's been a really great experience. Working in research and working on the things that you really find important, that you feel that can really make an impact, is something that really appeals to me and was something that I may have not had the opportunity to do without the fellowship. It's been a great journey so far, and I'm really thankful for the opportunity that I've been given.
My name is Samantha. I work in healthcare and social practice, which is our faculty here at Unitec. I came to New Zealand 23 years ago, and the reason that I came was I won a Florence Nightingale Foundation scholarship. They've been an amazing 20 years. I've been able to have some amazing academic and clinical opportunities, and to be able to give to this country that has been so welcoming of me, my husband and our family. I have the best job in the world. I get to teach about a profession that I absolutely adore. I get to teach subjects that I'm really passionate about. The research began from the curious questions that I'm really interested in and inspire me to think about ways to make a difference in my profession. One of our major considerations is to think about how we best serve the New Zealand population from the context of the fact that we're getting older, and we're getting older really quickly. We need to think differently about what that will mean for tomorrow's health practitioners. And then COVID came and we weren't able to upscale the research, and then the opportunity for the MB Fittinger Fellowship came up. From there, we've gone from strength to strength. We have undertaken so far 2 national studies. We've also done a national survey of student nurse perceptions of working with older adults, and we were amazed at the response that we had, and the opportunity to change the curriculum that we've got and the opportunity to make an impact on people's lives is right there. And we're so excited for this last year. The reason that the MB Fittinger Fellowship is so special for me is that the recognition that early career researchers come from a really diverse background, are all different ages and come to study at different times of their life. It has been an absolute honour and a privilege to have had an MB to fit in our fellowship for this year and into next year.
Salafalava, my name is Moeata Keil. I'm a lecturer at the University of Auckland and an MBIE Science to Fittinger Fellow. My research looks at how Pacific families make arrangements for their children after the children's parents have separated. My parents separated when I was very young, so I sort of have that personal interest to it as well, seeing all of the care and support and stuff happening around me. I went through my whole degree, and outside of the papers I took in Pacific Studies, I did not see a Pacific person, a Samoan person, Samoan mother, a Samoan woman, a Pacific mother, a Pacific woman. And I'm having to do a lot of that connecting work of how to think through these ideas or these big themes through a Pacific lens. I was halfway through my degree when I fell pregnant with our oldest daughter, Sophia. So I took a year off, came back, finished my undergrad. I completed my honors and started my master's, and then we had our second child, Carmine. The timing of it actually worked really well for me. I could combine studying with mothering, so I spoke in particular to Pacific mothers who were struggling financially. If they were struggling financially and they were entitled to receive formal child support, what was the barrier, what was the disconnect between their reality of struggling and them pursuing child support? Why didn't they want to pursue child support? So I started my PhD, which looked very generally at Pacific parents, mothers and fathers' experiences of family life after they separate, which has lended to my Te Whitinga Fellowship, where I'm looking not only at Pacific parents, but I'm looking at extended family members. We have parents and everyone talking about extended family, but we don't have the research. I now have a permanent position in the department I wanted to be in, and the faculty I wanted to be in at the university I wanted to be at. And I do believe that this fellowship had a huge part to play in that. It made me extend my research. Also, it shows the university that I'm doing research that matters and that people and institutions and government are wanting to fund. I've been lecturing since 2015, but on contract to contract that sort of rolls over, and you're not really sure what the next year holds. So to have some certainty, or to be part of the furniture is a really, rewarding and just really like, not I've arrived, but I'm kind of,
Kia ora, ko Mahonri Owen tōku ingoa. I te taha o tōku papa, he uri ahau no Ngāpuhi rawa ko ngā te tūwhare toahau. I te taha o tōku mama, no Uropi ahau. My name is Mohammad Rehan. I am a senior lecturer at the University of Waikato. I am a MBIE Science Fittinger Fellow. In 2008, I had the experience of going to South Africa. There was a lady who had gone through a few things that I had no ability to help her. And I thought to myself, well, that I wanted to help, I just couldn't. So from that point on, I thought whatever I do for the rest of my life, as long as it's helping people, then I could be happy. That's what I've been doing, trying to create these machines that can help people who are suffering from nervous system disorders or from amputations. The opportunity to do a doctorate came in. Between me and my wife, that was a big decision to put the next 4 years of your life into study, whereas I could have been working. But we together decided to do that, which I'm very thankful to her for. There was a lot of sacrifices she's made for our children and for our family, so I'm indebted to her for that. But I was successfully able to complete my doctorate, and that's when COVID hit. Everything kind of stopped academically. I was in principle offered a position, and then COVID shook that. So I kind of had to leave for 2 years. The opportunity to apply for the Whitinga Fellowship came, and I thought, why not? And then after successfully receiving it, I was able to negotiate a full-time job. So yeah, right now I'm lecturing and doing research and kind of learning how to navigate that as a Māori in a space where there isn't too much representation. Māori are a very beautiful and a very clever people, and I think that gets lost sometimes. The previous 2 years before coming back to the university in the tertiary space, I worked in Māori capability development. And it's not because Māori can't. We just need to do a better job in looking after them. What is the most important thing in this world? And the answer is he tangata, he tangata, he tangata. It is people. And I've noticed that wherever in the world I go, the answer is always the same. It's always people.
Kia ora, my name's Rebecca Campbell. I'm a scientist at Plant and Food Research in Motueka, and I'm a very grateful recipient of the MB Whitinga Science Fellowship. The research for my fellowship is all about epidemiological modelling for plant diseases. It's quite handy with COVID. We've had a lot of conversations about epidemiology and epidemiological modelling. In some ways it's kind of similar. We are trying to flatten the curve of the plant diseases. One of the things I love most about my work is it's so varied, and I get to go out into the field and talk to the growers and have that real connection, and I get to do lab work. There's just so much more to know, and everything you do find out, there's more questions that come from that. I guess I'm driven by that curiosity, and actually there's a lot of creativity in science of thinking up the solution that could possibly work. So I went to university in Christchurch. I did my honours in entomology and fragmentation science and community ecology with insects. Then I did my PhD with freshwater science, so I was a freshwater ecologist. I did some part-time jobs while I had my children, and to get into plant pathology, there was a job advertised here as a part-time technician. I thought that fits perfectly, I can do that. And then once I got into it, I realised actually I can bring in all my ecology background and my spatial ecology background and apply it to this new system, the pathology. In plant and food, we're constantly applying for funding from anywhere we can, really, and putting together a proposal has to be quite concise to apply for a fellowship like this. I had to check properly that I was eligible because I have had that time off since my PhD. I had the family break, and I think that's really, really important that that fellowship recognised that actually I was still an early career researcher because I had been working part-time. So I think I will carry on championing this area of research. I think it's really important, and already people are reaching out to me interested in the skills that I've developed or developing. So I'm quite inspired to apply for some funding that really connects internationally. I think there's a lot of knowledge that we can have from overseas. For example, organisms that we don't have here yet but might come here, and we can bring all that knowledge back here first and be ready, more ready at least.
Hi, I'm Sam Crawley. I'm a postdoctoral researcher at Victoria University of Wellington in political science, and I'm an MB Science Whitinga Fellow. I came to research a little bit later in life than some people. I was really worried about climate change and wanted to do something on that. It's a really pressing issue, an issue that a lot of us are worried about, but there's not enough being done about climate change. Politicians are not really putting the really sort of ambitious policies in place that we need to really address climate change. So I did a survey in Australia and New Zealand. I asked people all about their different opinions on climate change. A lot of people wouldn't expect to see that difference, and a lot of people might think, wow, climate change is probably more important in New Zealand than Australia, but actually, yeah, it's the other way around. Things like healthcare, especially in recent years, education, especially for people with children, and the economy and job security, cost of living, those sort of issues tend to be much more immediate to people. COVID kind of hit in my last year of my PhD. I didn't really have too many options at that point because there's still a lot of uncertainty. I would have had to have tried to pick up casual contract, or I may have had to considered leaving academia potentially, given the sort of situation at the time. So I'm really grateful that I've got the Whitinga Fellowship. So I'm the co-chair of Manuhanga, Wellington Postdoctoral Society. We're trying to help postdocs navigate the system, how to apply for grants, how to apply for promotions and pay rises and those sorts of things, and just try and build a community of postdocs, at least around Victoria University. Once the Whitinga Fellowship is over, still a little bit uncertain at the moment. I'd really like to stay in academia and try and ideally find a permanent role, probably at a New Zealand university. Being able to do research, it is a real privilege. I get to work on problems that you're really interested in and problems that I feel that are really important. There's no bigger thrill than collecting some data and then analysing it and finding out something that maybe not everyone appreciated, even like other academics.
Kia ora, I'm James. I'm a lecturer in the Department of Mechanical Engineering here at the University of Canterbury, and I'm an MB Science Whitinga Fellow. My research, or my interest, is in fluid mechanics. From my PhD, I wanted to stay in academia. I enjoyed the research environment and the freedom to sort of pick out and target things of interest to me, and I stayed in the field of fluid mechanics, looking at lava flow rheology, which is important for volcanic eruptions to be able to determine and forecast how quickly these lava flows will disseminate from the volcano. From there, I applied for the Whitinga Fellowship. So this was on a project of looking at the blood clotting, and it has an interesting connection to my work during my PhD. My PhD, I have worked on silica scaling and geothermal heat exchangers. So this is pipe flow. Our blood vessels are also nice pipes, and we have this plaque that builds up over time, and this is quite analogous to our silica scaling and geothermal. After finishing my PhD, I was looking at next steps and opportunities. So we have very, very few postdoctoral positions in New Zealand. So I applied to the Rutherford Foundation, applied to the Marsden Fast Start. So these were unsuccessful, as often is the case, but we kept trying. Fortunately, I was awarded this fellowship, and that enabled me to plan for a 2-year solid research project. And this has enabled me to make new connections with other organisations and funding bodies within New Zealand. So there's a new Centre of Research Excellence, Healthy Hearts for Aotearoa New Zealand. So their focuses on cardiovascular diseases, particularly within the Māori and Pacific Islands population. By engaging and collaborating with this group, I'm able to form networks and links with the community and clinicians to hopefully ensure that my more theoretical and modelling analysis can be applied and connected to real clinical outputs, and essentially, at the end of the day, save lives.
There's been some fantastic presentations, and it's always really hard to find a winner in a very, very short time. But before we get to that, we just want to play a small video from one of our supporters. Nishan Shantilaya is the Horizon Europe's representative in the Australasia region. They are based in, he's based in Australia, but he's just recorded a little video presentation that I'll play in a minute. Horizon, or Euraccess, as it's called, they have a small prize for the winner where they will help them to kind of give them some presentation tools so that they can do really well at the actual presentation in Berlin. But Nishan will talk more about that, so please play that video.
Hello everyone. Good afternoon to all of you. First of all, congratulations to all the finalists of the Falling Walls New Zealand competition and to all the participants who have endeavored to submit their Breaking the Wall ideas. It might sound like a cliche, but all of you who have qualified for the final are winners because you have accepted the challenge of presenting your research in an accessible way and in such telling times. Effective science communication never was this critical as it is today. The true value of science can only be achieved when it is communicated, and hence it is crucial that people know what is being done in the labs and how brilliant minds like you work to tear down the walls. I'm Nishan Sandile. I'm the Regional Coordinator for Euraccess Australia and New Zealand. Euraccess is an initiative of the European Commission that addresses barriers to the mobility of researchers and seeks to enhance scientific collaboration between Europe and the rest of the world. We provide free information about European research, career opportunities, international collaboration, and networking possibilities. These are really exciting times for New Zealand and Europe's research relation. A couple of months ago, the European Commission and New Zealand signed the association agreement on the participation of New Zealand to Horizon Europe, which is the EU's research and innovation program with a budget of around 95.5 billion euros for the period of 2021 till 2027. Now, this development entails that New Zealand researchers and organizations can now apply to Pillar 2 of Horizon Europe on equal terms as those from EU member states. To those who are watching us, if you are interested in learning more about Horizon Europe, get in touch with Euraccess Australia and New Zealand and let us help you. Consistently to our philosophy of reducing barriers to the scientific collaboration, we have decided on a long-term partnership with Royal Society, and this is to be involved with Falling Walls Lab in New Zealand. And as a gesture of our appreciation for your hard work, we will invite the winner of the competition to a science communication training and mentoring program, which will help them to prepare for the international context in Berlin. In addition, we will also facilitate connections desired by the winner in Europe should they want to establish contacts, and this could be to liaise them with research organizations in their field, connect them with any of the 600 plus Euraccess centers in Europe, or introduce them to prestigious European Research Council grantees or Marie Sklodowska-Curie fellows. Before I wrap up, I'm reminded of an apt quote by Sir Isaac Newton, who famously said, "What we know is a drop, what we don't know is an ocean." So I encourage all of you to remain curious always. Whatever the result of the finale is, keep up your quest for answers to your queries. With that, good luck to all of you, and I hope you all had a good time ahead. Thank you very much.
I'm back on. I'll now hand over to Professor Phil Lister, who will announce the decisions of the jury. So, got a microphone.
I have my own.
So I'll just first invite Winnie Switakowski and Brent up here so you can pass on the presents as well, or the awards.
Very good. Where would you like me to? Yeah, sure. So the committee would like to congratulate all the presenters today. You made our task extremely difficult. We would have liked to have given out many more than top 3 awards. There was a lot of debate about which people should get the awards. and you made our life very difficult. We would note that the standard of presentation from everybody seems to have increased substantially over the few years that we've been involved in this. So now it's fantastic, and again, congratulations very much. We do have top three, and I'll go from 3 to 1. I believe that's what I'm doing. So the third place winner is Kristin Bohm. So congratulations. Kristin's project on black soldier flies could turn what is a surprisingly large amount of waste into a resource. It was an excellent presentation, great answers to questions, and innovative use of the material. So extremely relevant to a growing population. So well done. The next two were very hard to differentiate, and we had a lot of debate and trouble doing it. But the second place winner is Tejesvi Patel, breaking walls in fiction using PVP. Congratulations. This was a really well thought presentation on multiple layers, really. So we thought very relevant to a world in which we're living, which in microbial infections are a growing problem and increasing around the world. Very well presented, really nice. Congratulations.
Thank you. Thank you.
And the first place winner for today is Debashree Roy, plant-based cheese. Congratulations. Very good. Well done. The committee really wanted to taste this cheese.
Please, you are all welcome to come to Massey University, and I'll have to taste them.
Sign of disclaimer.
We are looking forward to this immensely, this trip. It's an innovative and really useful project that could have real global implications around the world. Answered the questions really well, really engaged in the presentation, and a big breakthrough. So well done.
Thank you. Thank you so much.
Do you want me to take this and you
Wait,
take it?
what should I do? I'm supposed to leave it over here?
You take that and we'll look
Thank
after
you.
this.
Sounds good. All right.
Yeah, thank you. Yeah, thank you.
Thank you, Phil. Thank you, Winnie, and thank you, Brent, for your help here. I just have last two important jobs to do. First of all, I want to thank our jury. Really appreciate you taking your time to come here. It's a whole day out of a busy schedule, so thank you very much for supporting the event and the excellent early career researchers. So if you just please come up here, and we have a little koha for all of you. So if you just join me in thanking the jury for being here today. Right, the last thing I just want to say, a big thank you to our supporters, to the German embassy in particular, who started this and brought this to our attention. And I think we've enjoyed a long and very nice collaboration since the early days. I also like to thank Euraxis and Nishan, and last but not least, I'd like to thank the Ministry of Business, Innovation and Employment, who is the main supporter of this. And without their support, it would be very difficult for us to run this event. There will now be a reception in there in a minute, says Holly. Before we go to the reception, we'll just want to ask everybody up here for a big group photo. Out there, okay? But before you go out there, I'll just officially close the event here today by saying Hoki pai aiki o koutou kāinga. So return safely to your homes. Some of you might go straight home now. Some of you might go and enjoy the reception. I hope most of you do. But before you go anywhere, follow Holly's instructions to where we are going to take a big photo. So thank you very much for coming today.